So the another example I'm showing here is that the green one is showing that you can put the peptide and how it's going to treat the peptide. But what you could do also is you can design a compound as a like kind of we call a ligand, and you put it in that same pocket. So it can inhibit that aspartic protease cleavages. So you can basically stop the inhibition, and whatever the process of pharmacology is, where you can downregulate that. So this is the mechanism total. The aspartic proteases can chop the peptides. If you can design a ligand which can stop the inhibition, it will be useful. And this is one example I have just shown you, that multi-billion-dollar drug that's called darunavir. You can see here is in the structure, and how the darunavir does basically is for HIV proteins, and how you see this dimer and then the two sparkle that warhead, and the ligand comes and bind in the same pocket. So basically it's mimicking that transition state there, and then peptide cannot come and cannot be chopped, basically inhibition. And the interesting thing to know that Arun Ghosh is an Indian scientist. He was the M.L.K. He was the pioneering on that. He made a drug himself. So this is the total mechanism concept of the aspartic protease. I was highlighting here. It's going to be foundational work. Any question? No. So what we're going to take about this, if the aspartic protease is now we understand could be a very interesting target. Where it is associated is that. So I want to give one example of the blood pressure. How many people are in the audience and wherever is online, if you look around yourself, and you will find out in a family and the friends, someone has a high blood pressure. So how you maintain the blood pressure? So I'm going to give a little overview about your blood pressure. It's called the Renin angiotensin system, which is regulating whole blood pressure of your body. And then the angiotensin is produce the peptides, you see the protein, and the renin is the first enzyme that I talked about is a aspartic protease enzyme, which cleaves as a leucine versus the valine and generated the angiotensin 1 decapeptides in the pathways. And then angiotensin 1 decapeptides, which is here, is the further down regulated by here is another enzyme called ACE, angiotensin converting enzyme. This is zinc metalloproteases enzyme. It's cleaved that decapeptide to the octapeptide. So chopping here again. So you can see the reason I have shown the low pole, you can see and understand what's going on the rest. And you generated the octapeptide here, angiotensin second receptors here called 81 and 82. And then in the plasma renin activity feedback loop, whatever these things are happening here, it gave a message back to the renin, and the renin trigger. So it's just going on and maintaining your blood pressure. So any patient who has a high blood pressure is going through these drugs. Either the drug which we have called ACE inhibitor. You can see these are all generic drugs exist in the Indian market as well as across the globe. Any of the drug your doctor is going to prescribe with the blood pressure. And the second drug he gave you, this is the called octapeptide, which comes the angiotensin second 81-82 blocker. This is called ARBs. These are the main drugs you can see here, the multi-billion-dollar drug already. So what happened, your doctor will prescribe either first AC inhibitor or the second ARB, and sometime in a combination. This has some problems, some patient cannot handle it. So there's another issue. So the interesting part is here that look about the renin. Renin itself can be inhibited and then can maintain the blood pressure. But the problem is that this is a really notorious and challenging target to make a drug. So that's what we don't have. I'm going to tell the story about that. So many patient has an issue. Even a combination of ACE and ARBs, your blood pressure cannot be maintained. So still you go to the doctor gave the medicine and it still doesn't work because of only thing is giving the ACE and the ARBs combination. Along with that, sometimes the doctor prescribes the calcium channel blocker or maybe the beta blocker or diuretics. But still, some of the patient population cannot maintain their blood pressure. So you need something else, and suppose something like the renin inhibitor here as well. Anyway, coming back to the story and history about this. Renin, why is it so important? This is a great important and challenges about this. It was discovered, renin itself was discovered in 1974 when basically I was born and I was playing a gilli danda somewhere in Ramfur village, as the Dr. Ansari mentioned it. And you see one quote here in the William Greenlee, he was an ex-VP of M.L.K. at that time. In 1987, he wrote a review in the paper. You can check this review paper yourself here. What he says about the renin, that all the renin inhibitor of renin have been studied for over 2 decades. Only recently has a substantial progress been made toward potent low molecular weight inhibitor likely to become a useful therapeutic agent. So he's saying in 87 that it's challenging to make a drug. And since then, from 87 to now, I will put a cartoon here a little bit, that this renin is an aspartyl protease enzyme and help control blood pressure and maintain the healthy level of sodium and potassium in your body. So it is a really useful target. Why not make a drug now? So people tried to make a drug, but they could not make it. I just mentioned since in 1987, he is a VP of the Merck. He is doing that research that time, and still they have not made it. And this is the review. I put it here for you guys. What is the drug discovery? How it look like? Usually take a 10 to 15 year of discovery a drug. We start a pre-discovery in early space here. You can see we made a many compounds, 1,000 compounds, and then we go the further development, a preclinical, clinical stage, and eventually we develop one drug which is approved by FDA. For this case, is rather than 10 to 15, direct renin inhibitor, it took 40 years to discover one compound. And that is discovered by Novartis, is called renin, Tectona, and it is available in Indian market as well. So the problem for this, which I want to share here right now, although it took so much time, it is a peptidomimetic compound, it's a huge monster molecule, and is only bioavailability is 2.6%. So the most of the audience obviously not familiar, they might be thinking, what the hell is the bioavailability is? I'll share a little bit what is the bioavailability means that. The compound, the renin inhibitor were made after 40 years of hard work, and then we discover and it's making multi-million-dollar revenue for the Novartis as well. But what is the problem for this molecule is that so much 2.6% oral bioavailability. So I'm giving a simple cartoon to you guys to look at this. What does mean of the oral bioavailability? The fraction of absorbed into systemic circulation is the drug's called bioavailability. For example, imagine this is go to the Resilience, go to the outside of the market in the Balrampur, you see the counter, you see the medical store, you see this drug, this called Resilience. This is the aliskiren. In Indian market, generic is called Resilience. It's 300 milligram pills, huge monster molecule. You take oral administration by mouth. When you put in the mouth, it go to your guts, and then by a guts wall, it go to the portal vein, and the portal vein, all the stuff goes to the liver. Liver is then like a bodyguard, and he's watching that what is coming in and what is going out. Most of the stuff he chopped it, and then back to the systemic circulation and reach up to the whatever the renin target is to there. So literally, you see, out of 300 milligram, only 2.6% is reaching to the circulation. Means 7.8 milligram. The rest is gone. And where it goes to the feces. 292.2 milligram. It's just giving a rough example. It's useless. It's wasted. So what happened that this compound is still there, but the oral bioavailability is good. So if we can improve the oral bioavailability for this, it's very useful. Imagine the scenario you're taking like 300 milligram compound in your gut, in a stomach, got a stomach problem or whatever it is, because those are useless. The compound is only 7.8 milligram has been used for that activity. Anyway, so coming back to the story, how are you going to make this new and drug? So we start this called the structure-based drug design, which we have find out in the base program which we developed, that fragment-based. We got the fragment and put the NMR studies of the protein. You can see here, this is the one small fragment which we discovered initially, bound to the protein in the catalytic pocket, where is the aspartic acid here, so 222 here, and this is showing. It is engaging with that warhead, which initially I was showing that aspartic proteases. So it means the small fragment bind there. And then when we found that the medicinal chemist, that's what we do, based on our structure design and finding out the more efficacy, we designed this different compound, and it's almost 100 patents discovered in this area. And it took it 10 years to discover one of that important compound. And you can clearly see here in that base inhibitor, across the globe is not only Merck. Merck was making the same. That is our compound, which goes to the phase 3 for breast program, Alzheimer's disease. J&J has, AstraZeneca has, Lilly has. Most of that of big pharma companies have the base aspartyl protease inhibitor, and they were running to the clinical stages for the efficacy. So this is the example I'm giving. We discover those aspartyl protease inhibitor for the base program, not the renin. Then what we did it, when we saw this opportunities, that says we have that aspartyl protease inhibitor, why not we can exploit this for the renin? Renin is also aspartyl proteases. So that's what we did. We get the cluster of these aspartyl proteases and run in that our enzyme inhibition activity with the renin. We find out 82 nanomolar compound.
Excuse me, Tanwar. Can I request to the organizers to mute their phone, please?
Yes, sure. You're right.
Disturbing lot.
Background noise is coming. Thank you.
Yeah, please mute it. Please mute it. You unmute only when there is a need.
Thank you. Appreciate it. So we got the first renin inhibitor here, which is 82 nanomolar. You can see here. And now we want to obviously discover this drug, actual a drug which is going to be efficacious as human model. What we're doing here, whenever we the drug discovery, we first started the simple having a protein and in vitro that's called test tube thing, right? And then you tested that this is active or not. The activity is itself not is important because the most of the other profile is important. For example, there's a more sparkle proteases found in a body, like base one, cathepsin D, which is if you inhibit it, you get a problem in the ocular. If it's found in the eyes, you can be blind. You can have some other potential liabilities or issue. That's we call the selectivity. Whatever the other sparkle proteases in the body is, you need to select it. You have to selectively hit only renin. That's why you make a potential important drug. Otherwise, if you have other inhibition, that's it's not going to work on that because that efficacy comes from there as well. Anyway, so that was the story. But then we got a potent compound, but this has a poor PK, like same that we were talking about the aliskiren and the HERG liability. There is a cardiac toxicity we see right away, which is we call HERG. Well, this is showing that if you take this type of compound, this does not affect your heart problem. And then selectivity of, is there anyone?
No, you carry on.
All right. So we started this the discovery by a structure-based drug design. How are we going to do that? I'll give a real example here. This is the aliskiren in yellow. We soak in this the protein that we talk about, the aspartic protease enzyme. You see where it is binding in the catalytic pocket, and this is the compound which is initially we have. If we do the computational work, that's we dock together, and you can see how it is binding in the different pocket. If we clearly you see the S1 pocket, where is a huge molecule, which is a graphene here, and chop it with the S1 with the isopropyl. And that's what we did something. So the reason I want to show these is leveraging the protein structure and structural information as well as a computational approach, how we can solve this medicinal chemistry and guide the SAR. So chopping this and putting the isopropyl get a 50 nanomolar compound, and then eventually putting this methyl group, we see that they flip out of this S3 to S3 subpocket, and this improved affinity to nanomolar as well. So these are the few examples I shared with you. Obviously, traditional STR we did, we made a many compound of, you can clearly see from left to the right here, and we have a potential liability and issues as well. For example, the red part is showing again, this compound is really potent, but there is a problem of permeability. What the permeability means is that these are the terms we use for when we are discovering drug. But when you take it in the gut, it does not permeate in the cells. It doesn't go there. So it's used. So we have to improve the permeability or the AUC, which we're talking the bioavailability and the downstream. You see that how much compound we need. There's another example which you see that VT and another company at that time, they were also trying to work in like a sparkle protease inhibitor for renin. And you can see when they made it, the chloro component meta position here. And here in the S3 pocket, there's a chloro is showing here. And in our compound, if it's clearly visible, if you put the R with the chloro, it can improve that. So we borrow those information from VT compound and from 44 nanomolar to 2.2 nanomolar, only putting the chloro. This is the example I'm showing you how the protein and crystal structure can guide you to improve the affinity of your molecule for moving on the direction. So these type of the avenue which with the medicinal chemistry people approaches and we solve the problem is called structure-based drug design. And based on this structure-based drug design, we discover many potent compound, and finally we made this compound SP10, which you have shown here. This is the core structure which is binding to this aspartic proteases, and it is a center and flipping here. And you can see 0.6 nanomolar compound, all are green. There is no potential issue, and we can move on to the in vivo activities. And then we also did we call the tox study. What we did the high dose compound, you put the rat and mouse or even dog, monkey, and pig to see that if any the side effect is coming. That's what we do that very early. So first we started the minimum, that's the rodent, the rat and mouse, and we see that the enzymatic induction it was very clean till up to 100 milli MPK. So this is really good news. I've just showed you the story that how it evolved from one tiny fragment to actually
candidate which looks like really, really important. Now the question is this, whether it's going to be really reducing the blood pressure or not is only just making a drug design. That's the next step for you to do that. When you have a safe molecule, so before you come here, it almost takes 2 to 3 years to get this compound. The reason is that you want to make sure everything is safe before you go to the animal model, because you don't want to kill the animal and eventually you don't want to kill the human in the phase one. So that's why we go to those parameters, and that's the major challenge for the medicinal care to solve that issue. And it is really, really important for us to that. When you solve that challenges and a victory, you got a tool compound, you have to show the efficacy. The efficacy means whatever the disease relevance you have, whether it's working or not. For example, in this case, we're talking the blood pressure, whether this compound is going to reduce the blood pressure or not. So, for example, mouse doesn't have the high blood pressure. So what we did is basically we made a humanized mouse that is called double transgenic rat or mouse, which has a human brain incorporated. And when you have that, it elevated the higher blood pressure. So for example, here, look, the L-screen, which is the actual drug in the market right now, you take a 10 milligram and you see the lowering the blood pressure right away, right? And you take a 30 milligram, it's also lowering the blood pressure because the bioavailability is not, it's a very, very potent compound. Took our compound which you made, only even a 3 milligram versus a 10 milligram, very should drop of the blood pressure right here. So this is a clearly demonstrated the efficacy that with the target, when you hit that renin inhibition and you have enough compound exposure in a blood, you can reduce the blood pressure. So this is the one of the example I shared here. What I want to bring it here, this is a nice note. So when I told about the Will Greenlee, he was a VP for the Merck that time in 87. When I was working with this program with my boss, Brian McKittrick, he was a VP again to the Merck. And what he said again here in another review, after 27 years, he wrote another letter in this paper, Annual Medicinal Chemistry Review, and this is the page number 20, what he wrote, that creative lead optimization, Brian McKittrick and Will Khan were able to discover potent selective renin inhibitors such as inhibitor 6. Having struggled with the peptidomimic renin inhibitor during 1980s, it was particularly gratifying to have this success over 25 years. So this is a really nice story, end up from one, when it was paid and now, and he is still watching that story evolving. Almost 25 years for one particular target. This was very, very nice. It's very rare usually. We don't go that far basically. If any target is not, nowadays we are not patient. 4 5 years, let's move on. But this is a very nice story. You guys might be thinking that what is the chemistry? He is a chemist and he's talking about how. So how we made it look simple. If you take the isoproper ketone here, you do the Jonathan Elman chemistry right here, and LDA chemistry, inertia selective here, making the primary amine reduction here. You can see reducing by dioxane and SCl releases this, and then cyclization give this core structure. This core structure further reduction of this benzylic position give this biometh snowball. You got this core structure. Hydrolysis of the ester with the acid. And with this acid, you have your amine fragment and couple it. And that's your drug. You can scale it up tons and whatever you want. So this is the medicinal chemistry route, and it still is very good for the process as well. So I think I will stop for a 5 minute. You guys have any question here? Otherwise, I'll go to the second story. Anyone?
No, you carry on.
No?
You carry on.
Tanvir, I can get into.
Okay.
Yeah, see, this is Harish actually started my time when I was supposed to be there. We both are in US, that's a horrible time anyway. So, the question that, why you think that, sensitivity is more important for renin inhibitor or specificity in the sense because renin is also very important system in our body for human physiology. If you inhibit the renin at very high sensitivity, and you are trying to improve it, you think it will allow the system to run in our body? I think low sensitivity was possibly just to see that, you can still survive while controlling your BP, etc., which you mentioned. I'm not from the field, I just thought of
No, obviously. So that's another good question. Everything what we do in the medicinal chemistry has a check and balance. So you cannot shoot it up. That's why we need a doctor to get whatever. You cannot take as much as you want over the counter. So it will define it your blood pressure regimen, what is going on. whether there's anything working. So imagine the scenario. It is a row, as I mentioned, is a feedback loop. Whatever is going to happen, even you're innovating here, this is going to stop. If you're going to stop, already ARBs are going on now, they're going to stop here. So it's a balancing act for the nature. So body is balancing act. And that's why based on that condition, that much amount of the compound will be given. That is important. So it's a great question. It's not that randomly you can take whatever you want and go and have your blood pressure. Obviously not. It will be properly checked.
Right, okay, go ahead, maybe. But my question was like this only, that why do we need to have very high sensitivity, that is efficacy which you mentioned,
Yeah.
rather than selectivity?
Got you. So the sensitivity is not a proper one, but we can say that EC50 is more potent. Is that if you don't have that, then off target is also a problematic. What we call a therapeutic dose, right? When you have a therapeutic dose and window, more the potent compound you have to less the compound to use it. You can use them less sensitive or less IC50 compound also a drug, but there is a therapeutic window which we are. I was telling the mitigating other cardiac liability or other toxicity profile. So that's why minimum compound, more the potent, better is their profile. So the second thing I was going to share here with the spartal proteases, when we discovered these spartal proteases, I was very excited to see that in the malaria, there's a one paper came in the Professor Alan Cowman, which in the Nature he published that he discovered that there's another spartal protease is found in the Plasmodium, Plasmodium vivax and Plasmodium falciparum. That is important for their proper growth. So when I saw that Nature paper, I came with this plan and an idea. You can see this was published in that. I have sent the link here if anybody want to see that. It's already BioWinter Global Health has highlighted my picture and name. What he said here, I just want to see, you can read the detail, but here is that in that at the US-based pharmaceutical company Merck, known as MSD outside the United States, Canada, Dr. Chanwir Khan recognized that the company had a mini drug like aspartic protease inhibitor, and it's a chemical library that could potentially impact the replication of malaria. So what we did basically, I was very excited, but the problem for the malaria in the Western world, we don't care about the malaria. It's not a profitable business. It's a poor people like India, China, and the third world. So that's what it's no money for making. We're talking about the billions and millions. Here is simple. We need a $1 dose for treatment of the poor people. That's the guidance for World Health Organization we have. So what we decided that we can go to the external partner. I went to the Wellcome Trust and asked them if you can provide the funding, you can do this one. And then we got the initial 200K of dollar, and basically 200,000, not the K, I'm sorry, the here. And with this, we have a post-RN generated initial data. And with that initial data was exciting. We went back again and got Wellcome Trust Seeding Drug Discovery Award. There's a $4 million, around 30 crores that. Once they achieved that grant, based on that, we started the discovery program here in identification of this malaria. And you see what we did. These are the spartal proteases we have made it initially. We did the clustering of that, and we went to the Australian counterpart and helped them to screening those component Plasmodium falciparum in vitro assay with the cell lines. And we find out there's a one drug hit. Out of those 32 compounds, we got a really one drug hit. And you can see this is something look like that is the color what the pinkish color it has. This is the compound which comes from the malaria, the renin inhibitor. You can see here. So it was very excited. It's a 4.6 nanomolar which is hitting. So that since the question was before that the sensitivity, you can imagine now, even one phenyl group we're putting here, it wiped out the renin inhibition activity completely. And now is another portfolio. You can see that for a plasmodium. And then you can see initially when we got that plasmodium species and infection, this is the film tool compound. It was really, really potent, but they have a poor properties. The PK, that's what I was talking about. So what the team did basically, I was not part of that. We have the money when we hired the teams and the lead optimization discovered this compound is a tool compound WM382, which has a really, really potent compound, and it advances that further. And this is we published in Cell and Host paper early in 2020. I don't want to share this is the cartoon, you guys are very much familiar, but I want to share this tool compound. How important is this? You can see the different dose regimen. You can have exposure. This is called AUC, in a PKPD, that this much compound is already on board, so you can kill that malaria parasite. And This is really remarkable. The reason is that even one single dose, you can clean the malaria completely. You can see here. This our compound is a 1, 3, 10, and 30 MPK. You take the 30 MPK, it's completely wiped out the malaria in one single dose. What we did basically confirm back again. This is the in vitro we're talking. We went back to the animal model. How do the animal model? So we have a mouse and the rat. Obviously, those guys don't get that. The malaria infection as well. So what we have to do, we have to humanize mouse back again. We made the humanized mouse model for this one. You clearly see here, this is the infection in the liver, which we're monitoring for that. And this 52 or 65, this is the control. No one is going everywhere, the malaria parasite in the liver as well as in the circulation. And here is a 20 milligram dose, 100 milligram dose, completely clean the malaria there. And now, interestingly, this compound is moving on to the phase 1 now. Everything is passed, not this compound, but other compound which I have not disclosed here. The compound is basically clean. What we say the phase 1 is this is the animal model we did it initially. Before we go to the human, once we clean the compound with everything, whatever the liability is there. Like I started the program in 2013. Now it's talking 2024. Already 11 years gone. What happened now? The compound is neat and clean now. So we go to the phase 1. Phase 1 is called a healthy volunteer, like me and you, anybody want. We want to take the dose and compound and see how much the therapeutic dose we're going in the blood, and we're monitoring that going on. So once the phase 1 data comes positive, we will go the actual patient population who has a malaria and going on. So this is a very, very exciting evidence. The reason I'm saying this is a very exciting because it's a charitable work for us from the M.L.K and from all of us, but it's going to save a millions of patients, especially the poor world, right? Like where there's no profit and businesses, nobody is going to do it. And it's like one single dose and clean the completely malaria out. Think about it. I had a malaria myself when I was in India. The chloroquine which I had taken, it was shaking my whole bloody body. So that's what the excitement. I want to lead this program from myself. And thanks, touch wood, that this is moving very, very well. And hopefully in a few years, this will be in the market. That will be remarkable, especially the pregnant women and then especially children. So one thing I want to just show you here is a cartoon. You can see that what's happening, but I will not touch upon that one. I want to just say thank you about something here. Whatever I have shared and talked about this, remember one thing about the drug discovery. There is no personal contribution here. Always is a team. It's not a team even, it's a village, it's a city. We can imagine 1 year, 2 year, and 10 year a journey going on and many, many people involved. So like even when we see about that, someone is talking, I did. It's no one did call I. It's the whole team here. And you know what the individual person can take the contribution that this is my program and my thing. It's an entire village and it's take ages. And many, many people has involved in the, and basically contributed for the discovery of art. Thank you. Any question?
Thank you, Dr. Tanvir, for your excellent piece of work in the field of.
Dr. Ansari, can you hear me?
Yeah, I'm hearing. The voice is audible?
Hello.
Hello. Dr.
Yeah.
Tanvir,
Any question?
whatever questions the audience have, they will email you, please. We are running short of time.
Sorry, what is the question?
If any question the audience have, they will email you.
Email? What is that?
They will mail you. You answer in the mail.
Email you. I mean,
Oh,
no live. You can do offline. I mean,
yeah,
beyond
sure.
after.
You have my email address. Yeah, please
Yeah,
feel
yeah.
free. That's what I share everything.
We'll
And
share
with
the
respect
email.
to this program, about the other program I have shared, anything. For me, MLK, it's unfortunate moment that I'm talking from online. I would love to see and our folks and colleagues around there. So please feel free to communicate me, but not the phone. Just mail, please. Right? That's the only thing. So my email is there and my LinkedIn is there already. You can get it to me. We should get connected. Any help, any suggestion, I'll be happy to help you guys.
Okay. Okay. Thank you, Dr. Tanvir.
Thank you. Thank you and have a very good day, guys. Bye bye.
Before I introduce our next speaker, Professor Haricharan Misra sir, we are extremely sorry for delay due to technical issues. Professor Haricharan Misra sir, retired as ex-head Molecular Biology Division, Baba Atomic Research Center, Mumbai. Currently, he is working as head Center for Multidisciplinary Unit of Research on Translational Initiatives, Gandhi Institute of Technology and Management, Deemed University, Visakhapatnam. Professor Misra was visiting professor in Toyo University, Japan in 2019, in Harvard Medical School, Boston, USA in 2012 and 13. He was also visiting professor in the University of Texas, Southwestern Medical Center, Texas, USA in 2011. He got postdoctoral fellowship from Rutgers, New Jersey Medical School, Rutgers University, New Jersey. He got his PhD in 1988 in biochemistry and microbiology from G.B. Pant University of Agriculture and Technology, Pantnagar. And Professor Misra has various awards, fellowships, and recognitions. Some of them are as a Fellow of National Academy of Science, India, Fellow of Indian National Science Academy, Platinum Jubilee Lecture Award of Indian Science Congress, and there is a large series of such achievements. He has published more than 100 research papers, and the topic of his today's talk is Radioresistance in Bacterium, Roles of Chemical Repertoires. Over to you, Professor Misra, sir.
Can you hear me?
Yes, sir.
Am I audible to you, please?
Yes, sir. You are.
And can you see my slide?
Yes, sir. Your slide is visible.
Is it moving? Can you see next slide? Hello?
Yes, sir. Yes, sir.
What are you reading now? What is the next slide? Is it moving or no?
No.
So, what, do you see next slide? Can you see my third slide or no?
First slide is sir.
Only picture.
Yes, sir.
Okay, so that's the thing. Can you see now?
Yes, sir.
So, is it enough if I go in the editing mode? Because it seems full presentation is not accepting. My contents are visible to you. It's
Yes, sir.
reachable.
Yes, sir. Carry on your
Okay.
presentation, sir.
Okay, so I think I will go in this mode only. Let me start it. So thank you, Professor Ansari. I think I missed it. Personally, I would have been there. And at the outset, I would like to thank to Dr. Mishra and Dr. Pandey for reaching you out. You know, I did my MSc in 1983 from M.L.K. college, and since till now I'm not being able to live without a single day without remembering this degree college and this actually academic institution. And this slide is particularly to reach to our students and then younger people that how lucky we were to be part of this M.L.K. postgraduate college. And very recently I met to Dr. Afroz Ahmed. I don't know how many of you would know. He is actually a judge in the National Green Tribunal. And he came to our university and I had interaction. And we had a beautiful memories of those days, that how important and how renowned the faculty we had in M.L.K. degree college those days, and M.L.K. degree already has produced large number of alumni. They are around. Just now you have seen Tanveer. I think he is one of them, but there are many like us. So I always try to recollect my early days when I was just a village boy going to this degree college and started my life. I did my BSc in 1981 in biology, botany, and chemistry. I have a lot of incidents and memories when I was in the practicals for biology, botany, and what kind of I think inputs or the mentoring was done by the professor those days. MSc organic chemistry in 83. I think that was the turning point for my entire life where I'm sitting today. From there, I moved to Pantnagar, GB Pant University of Agriculture and Technology, and I did my PhD in biochemistry and microbiology. You see, there is a continuous change that I made, and then in Bhabha Atomic Research Center, I joined in 1990, and I spent whole of my time working in molecular biology and biotechnology aspect. So this is a kind of this slide is not only to see in a corner when I am sitting among my colleagues. I think all of them have done very well and they are around. Uh, the most important thing is there that there is a continuous change from geology, botany, chemistry, organic chemistry, biochemistry, microbiology, and then finally doing a lot of work. I did a lot of work in molecular biology, which actually decorated my biodata, which I had never thought that will be such kind of thing and will be spoken. Recently, I was in M.L.K degree college and I delivered a lecture after my superannuation. I thought, let me start doing some academic activity. And finally, I got connected with all of you. Thank you so much for actually inviting and for the students, particularly I mean, these faculties, if I spend time, I still carry a lot of memories. I have never found such a renowned faculties what we had in M.L.K. Postgraduate Degree College, Sir B.P. Singh, Sir Shukla, Sir Pandey, Sir S.P. Singh, Srivastava, all of them, and then they made strong foundation for us. So this is the degree college that students should actually take a note that you are being given a strong foundation in the college level and then the sky is the limit for you. Please don't give up. We have the ambitions to move out and occupy different places. I'm sure we still can do. I had a tough time in deciding and accepting the invitation from Dr. Pandey because it's a chemistry conference and all my work has been in the molecular biology. But I thought let me speak a component that we have been doing in the area of fundamental research by using. So I took this topic. I was part of the atomic energy establishment for 34 years. So radio resistance and the gamma radiation, they have been the cup of tea for our research throughout of my life. So I picked up a topic, radio resistance in bacterium. The roles of chemical repertoires has been spoken. And the work that I'll be presenting is still belongs to this red pigmented bacterium that we have been working, my lab have been working, and then this is the beautiful landscape of Atomic Research Center, another excellent place where I saw I got a lot of foundations to grow about it. For last 1.5 years, I am actually after my superannuation at the age of 60. I spoke in classified institute to continue, so I had to move, and I am grateful to this GESA University, very good university for last 40 years, and that has picked up many of us actually from different institutions, and we are collectively contributing to grow the research ecosystem. Under the purview of the NEP 2020, in fact, I wanted to reach out to uh, the principal and management of M.L.K. College also that we should have a strong. I mean, we had already, but we can get a better, strong strong orientation in the research program, and I would love to be part of developing such curricular activities. Let's move to give some feeling of the gamma radiation that will affect how our biological system affects it. Gamma radiation is very relevant kind of source in our, for human being, for society, because radiotherapy is one of the very important and effective treatments of the cancer, and it's going around the world. So we have been actually studying the effect of gamma radiation on biological systems and biological response to gamma radiation for a long time. And you just this slide is actually to give you a feeling that how human cells responds to gamma radiation. So if you try to, my talk will have a mixture of the chemistry, information, some science, discoveries, kind of thing in the area of fundamental science. Please bear with me. So here, what you see here, you actually see that the any biological system, they actually have the response mechanism to gamma radiation exposure. And when the cells are actually being looked, whether they survive, what are the doses at which they survive, then you end up of getting a response curve which you can see on the left hand side. And this response curve actually tells that what dose of gamma radiation can kill the majority of the cells. So the dose that can kill the 90% of the cells is actually called D10 here. And in human cell, just remember this figure, then you will appreciate the system that we work on. D10 is 4 to 6 gray radiation. So by giving this 4 gray gamma radiation to the human cells, to the cells of our fingertips, we can kill 90% of the cells. And this particular information has been used for using the radiotherapy in cancer cells, etc. Now, how do you monitor? This is for students particularly, that when cells are exposed to certain genotoxic agents, gamma radiation, physical, chemical, damaging agents, then you do a commit assay. This is a very simple assay, can be done in any college and in any laboratory. Actually, there's the established protocol where you can do everything on the other rows and the slide to monitor that what extent of DNA damage has occurred. And by giving 5 gray gamma radiation, you can see the tail which is coming out. Comet is a literal name. It's basically Puchhara that we say in Hindi. So in this case, the length of the tail actually it tells the extent of the damage. So by giving 5 gamma radiation, when 90% of the human cells die, you can see extensive damage of the DNA, and such kind of actually technique and experiments can be done in the laboratories without much expenditure. Now coming to the topic that actually fascinated us, and I'm sure after listening this talk, many of you will have several questions. That we have a group of bacteria. They are called actually dinos. We name it. Basically, full form is they called dinococci. A large number of species of dinococci. Bacteria. I'm sure that everyone is aware of it. That any branch of science you are. So you can see whole lot of bacteria. They actually belong to the family of Dinococci. And if you see the response curve here, the survival on the X, Y axis and X axis, the dose of gamma radiation, unlike the last one, what you see here that the x-axis was in centigrade, that is in the gray level. Now here what you see, the x-axis has the kilogray level. So we have a bacteria that even up to giving 6,500 or 7. Hello, can you please mute it? Okay, so you have large number of bacteria available in nature that by giving up to 6,500 gray of gamma radiation, which is nearly 1,300 to 3,000 times more than the human cells, and then these cells they don't die. Almost the viability remains 100%. So these bacteria they have eaten, that is the dose of the gamma radiation that can kill the 90% of the cells. They have nearly 10,000 to 15,000 gray like 5 that you see in human system. This bacterium actually, I mean, all these bacteria, they have a lot other effectors also. I mean, for UV radiation tolerance and desiccation, that is by drying the pellet, you cannot kill this bacteria. I mean, these bacteria under this condition. The most important thing which is directly relevant to the my talk today, particularly for the audience who have major chemistry background, the oxidants, we have hydrogen peroxide, which is actually a part of the reactive oxygen species or oxidant in our body that is being used for a lot of disinfectants, etc. And this bacterium actually can tolerate 40 millimolar hydrogen peroxide if you convert percentage it to the 0.12%, which is more than the concentration of hydrogen peroxide used for disinfections in our health sciences, etc. The point here that when E. coli, another bacteria which you can see here, when this bacterium can be killed by 1 millimolar hydrogen peroxide, this bacterium says bye bye. It doesn't just bother when the hydrogen peroxide concentration treated is around 40 millimolar. So with these questions, actually, discovery or identification of this bacterium has surprised the scientific community across the fields. And they all started thinking that how can a robust organism evolve and stay in the system or in the nature and that can kill it. So many questions were happening and plus a lot of hypotheses. To cut the story short, when the bacterial cells are exposed to 6,500 K of gamma radiation, this is the dose of gamma radiation when there is no killing of the bacteria per se, you can see the entire genome is fragmented in the twinkle stars kind of pieces you can see. But then when if you leave this bacterium in the same medium for 24 hours, it actually reassembles all the fragmented genomes into its intact thing. And this was actually was demonstrated in the University of Wisconsin by David Schwartz, and he named it in the Times Magazine article that Dinococcus, the ultimate DNA assembly machine, and this bacteria actually found a place in Guinness Book of World Record as one of the most toughest organism on the earth. We did a lot of research to again to reach out the message that by giving certain dose of gamma radiation at zero time, let's say cells are exposed to 6,500, and this is the genome pattern. If you analyze the total genogenetic material of bacterium, you can see that the unirradiated, this was the pattern, but after irradiation, this gets fragmented to nearly 200 double strand small pieces the entire genome. But as I said in the beginning, that if you leave this bacterium in the same medium, then you can say little less than 8 hours time, the pattern of the genome is looking almost similar to unirradiated cells. And when we check for the fingerprint, a mechanism is actually a technique to see whether this reassembly has made any change in the pattern of or ligation or the repair process. You can see almost the same pattern is recovered, suggesting that there is no genomic rearrangement happens. And all the processes for molecular geneticists, particularly biologists, have been very surprising because E. coli, if you see, it cannot tolerate two double strand breaks, while this bacterium doesn't bother when 200 double strand breaks are produced. So our the research in my lab actually, I started roaming around this particular phenotype. This is again a slide more for students and the people that gamma radiation, which is ionizing radiation, is actually have two different type of effect. One is that when the cells are exposed, any life forms exposed containing the genetic material, it actually damages the DNA by direct fragmentation of the genetic material, that is genome we call it. And the second mechanism is there that through radiolysis of water, where bacterium or organism they live under, or they have the aqueous environment in the cell. And then what you see that through radiolysis they produce large amount of reactive oxygen, especially like superoxide, hydroxyl radicals, and then they have a lot of mechanism. So, and then these reactive oxygen species, they eventually damage it. The proteins, lipids, DNA, RNA, and everything in our living system. Now, the question that I am actually today I am linking to this talk is that this the entire process is we call it oxidative stress effect because the reactive oxygen species they are actually oxidants and they produce this kind of situation. So then what happens that what you see here, these are the different things that my lab has been doing in our atomic research center. I am then continuing in my new place also on the different questions, the mechanisms that how Dinococcus actually tolerates such a high dose of gamma radiation and the questions that we had around DNA and break repair, it's a complete molecular mechanism which is very tough to deliver and understand in short period of time. The other is the oxidative stress tolerance that as you can see from the previous slide that ionizing radiation they produce huge amount of reactive oxygen species, nitrogen species. So how can they actually tolerate it? So out of several things, the fifth topic that oxidative stress tolerance, and I thought of telling you the remarkable repertoire that this bacterium actually has it to control the oxidative stress tolerance. This slide is actually a representative. I have got most of my representative data to reach you out, not in a lot of details. But here, this experiment which was done, I think you bear with me. I'll try to reach you out to the younger people. See, the question that was asked that when this bacterium is exposed to gamma radiation is producing lot of ROS and reactive nitrogen species, and how these dangerous or deleterious molecules they are actually touching the protein, DNA, and everything. So protein DNA component I have showed in previous slide that it removes it, but we still had a question that how proteins are being actually protected because even if proteins they are the ultimate building blocks and master of our biology and everything. So, but then if proteins gets inactivated, then what will happen? And this data was a big surprise. The experiment was done where three, four bacteria were picked up. The PP, Pseudomonas, E. coli, Thermothermus, which is the sister of Dinococcus radiodurans. And what was done? The cell-free extract was prepared ultrafiltrate. Kill the cells, then you pick up the entire extract from the cells, and you produce the ultrafiltrate and then these ultrafiltrates were incubated with a total protein of E. coli, and you can see the experimental design that I'm trying to reach you out. That you have 248 gray of kilogram of gamma radiation exposed to three samples which you had it, and one sample did not have this extract, and then other samples they have the extract. So when you do this kind, you have total proteins, then you put all the chemicals and molecules, the small molecules which are present in the cell-free extract of the bacteria or in the cell of the bacteria, and you expose with the gamma radiation, and then you monitor the damage of the protein. When gamma radiation exerts the oxidative stress, then some of the amino acids they actually form the carbonyls, and these carbonyls are the signature of the protein damaged by the ionizing radiation. So when this was actually we did this experiment, and what you can see here that when the cell sap or the component of the cells were taken from pseudomonas, which is not radio resistant, and we gave the increasing dose of gamma radiation, you can see the extensive damage of the protein, which gives the color formation blackening here. Same thing happens for the E. coli, which is normal bacteria. Same thing nearly happens to the thermos, which can grow at very high high temperature and is being considered a sister bacteria of Deinococcus. But in Deinococcus radiodurans, you can see this entire lane looks very clean, suggesting that this bacterium has got the molecules, that is small molecules, these are chemicals, which can actually protect the proteins from the gamma radiation damage. I think that was the conclusion. The entire thing was the abstract was picked up and a lot of metabolite profiling and metabolomics was done. And eventually we reached to the conclusion that Deinococcus radiodurans DR stands for Deinococcus has got a huge amount of manganese phosphate. One of the component was predominantly found, that is the manganese phosphate derivative, while others they don't have it, and this was directly implicated that it is because of the manganese phosphate, the proteins of this extract of Deinococcus radiodurans to protect the proteins of E. coli from oxidative damage. And further characterization was done, and this particularly work lead to a different direction where people started thinking that manganese phosphate could be an antioxidant or a compound which can protect the protein. And subsequently, some French Atomic Energy has come out with some formulations where they talk about something Dino product or lots of things started happening. So this was one very important component I thought of saying that a bacterium, one picks up this bacterium in the laboratory and try to do some lot of chemistry and identify the molecules, you may end up of getting very good quality molecules. This is another slide that we did in collaboration with the universities outside DRC in the India. And we did the metabolomics of Deinococcus radiodurans. This was not selective one. It was complete open research, was done open-ended research. And what you can see that here is a bacterium in the middle. You can see in the petri plate Deinococcus radiodurans. And here I have shown only 5 molecules that we could discover from our metabolomic study. And the one here you can see that bacillithiol is a redox modifier which can actually control the oxidant effect in the cell, which is already known. Other molecule was found to be indole scaffold, and the molecule a system, a bacterium or any biological system that contains the indole scaffold gives the tail-to-tail indication that this bacterium or this system might be making the anti-cancer compounds, etc. And these are the some of the predictions made that possibly this bacterium makes a lot of inhibitors, etc. for lot of enzymes which are medically very important enzymes. The next one is a dinogenetin. It's a beta carotenoid derivative, and look this structure. Dinogenetin can be extracted from this bacterium that has been found to have the antioxidant anti-cancer activity. But so far we have not been able to get the biosynthetic process of this particular compound, which opens a lot of opportunities for the our synthetic chemists. If they can get this compound, it will become the $1 trillion, as Tanvir was talking a couple of times, that multi-billion dollars. Okay. The other compound here you can see the peptide is also inhibitors. Some of these molecules were already identified in the lab, but I am being the molecular biologist. We took a particular route. Some studies we have done on the pyrroloquinoline quinone, a natural compound isolated from this bacterium, and I'll be actually taking you a couple of slides. So what we did what we did, we actually checked the activity of the PQQ. Am I audible to you, or I am only going on?
Yes, sir. Yes, sir.
Okay. So what we did that with pyrroloquinoline quinone, actually we check for reactivity whether this compound. I'll show you the structure. I mean, in the last one, I'm sure all of you know better of the chemistry. If you look this molecule and look the structure structure and electron density of this molecule, actually, I made use of my knowledge from the organic chemistry that I learned from M.L.K. college. I looked this molecule and I predicted that this appears to be a molecule that can be an antioxidant, and then we started moving on that. So when we did our initial experiment, we compared the reactivity of PQQ with the reactive oxygen species, and we checked the hydroxyl free radicals, superoxide radicals, and oxygen radical here. I mean, free radicals and oxygen radical, singlet oxygen, and it compared with the Tronox, the commercially available antioxidant, and aspartic acid, which is vitamin C. And what we found that this molecule actually is having the rate constant, etc., very close to some of the known antioxidants, and this is given to the neutralization of the singlet oxygen. So this way we actually concluded that the PQQ can be an antioxidant molecule, and we published this work in 2004, way back in the beginning of this story itself, in a very reputed journal, which is Waves Letter, actually published the novel findings only. So this is something which was going on, but a lot of work was done about PQQ, pyrrolophenolene quinone. Just to cut the story short, it was the first time from my laboratory, which we reported in 2003, that this compound can be antioxidant, and if you use a very higher concentration, this can act as a pro-oxidant also. And this was later, I mean, after that, this field of science in PQQ exploded like anything, and mitochondrial activity was shown by heat. Then somebody came forward and said that this can be a member of the B group vitamins, and lot of things was happening. And today I am glad to say, yeah, hello. Yeah, just few slides, couple of slides are there. I'll just stop it. So today I see in the when I go to Costco and etc., I see the bottles are being actually sold with the PQQ having a lot of potential application. Nevertheless, but this science grew very beautifully, and I was actually moving in a direction to prove PQQ etc. as a pro-oxidant and oxidant. But science, this has a hypothesis, but it should never be frozen. I think I have a couple of examples to tell you. When I was trying to provide all the evidence that PQQ can act as antioxidant, and my boss started telling in DRC that we can actually transfer the technology patent and everything. But as a fundamental scientist, I did not go in that direction. What I thought of making that this particular PQ2 compound is being produced in bacteria having 5, 6 enzymes, which is shown A, B, C, D, and E. And the PQQE is a regulatory enzyme. If PQQE is not there, then PQQ cannot be made here. You can see the pathway. So we generated bacterial strain. One was the wild type bacteria, that we call it non-engineered, that is something which doesn't have PQQ. And then we have made the E. coli that is making the PQQ from Deinococcus radiodurans. And what we did when we checked for the radiation response of these bacteria, which is biology, something which you have heard in previous slide, we did this experiment at different UV fractions, UVA, UVB, and UVC, different components of UV radiation. Data shown here is actually a remarkable turn in my science, where when you expose the cells to 254 nanometer of UVC radiation, which does not have a major component of oxidative stress-based killing, you can see these bacteria which are making PQQ that can become very high tolerant to gamma to UVC radiation. This kills the cells rapidly by DNA damage and very little to oxidative stress. So this was the first time when we started reporting some unorthodox findings about this molecule, that this molecule is working not as antioxidant only, it also works, has the metabolic effect. We went to Dinococcus and then we deleted this PQQE gene, which you have seen. I made the E. coli making PQQ, and we created the mutant. Mutant in the sense for in the popular language that this particular gene was deleted from the genome of this bacterium. So now this bacterium can grow, but it cannot actually make the PQQ. So and when we compared with the PQQ compound, HPLC profile and wild type and mutant, we could see that when you delete this gene, we have been able to abolish a peak that was corresponding to the pure PQQ. So we got the mutant, and then what we did, we checked the effect of gamma radiation, something which you have shown in the beginning, I have shown you. And what you can see that the bacterium that doesn't have the PQQ, that is basically PQQE mutant, has actually become sensitive as compared to wild type, which was highly resistant. So this gave some indication that yes, PQQE is actually doing some meaningful job in this bacterium. We did the pulse field gel electrophoresis, again some one slide that I had shown in the beginning, and you can see that the mutant that doesn't make the PQQE in Dinococcus cells, which don't make the pyrroloquinoline quinone, this chemical actually has become defective, could not do the reassembling of the fragmented genome, which was actually beautifully being done by wild type bacteria, which actually had the PQQ. So from these, we reached to the conclusion that PQQ is actually having some role to play in the metabolism per se, not in the protection of the micromolecules by neutralizing the ROS, but also in the correcting the damage that has happened in the cells. I have only two, three slides, but just pay attention, maybe for your information. We picked up the bacteria, we deleted couple of genes, but very summary slide that I am getting. And you can see there is one protein, it's called DR2518, which I have named it. If you see this response curve, in by deleting this particular gene from the genome of this bacterium, we could make this organism hypersensitive to gamma radiation, unlike the wild type which was showing extreme resistance to gamma radiation. When we look this particular compound, this molecule, this protein, what we found that this protein actually has got the kinase, that is protein kinase kind of domain, and it has got 7 sites where PQQ, the chemical compound, a molecule, a small molecule that was identified from this bacterium, actually has the interaction sites on the protein at the C terminal. And when we dock it by doing computational analysis, you can see that all 7 motifs, they actually folds back and create a cavity where PQQ could be docked in. So this gave some feeling that PQQ is actually working through this particular protein, and whole lot of research for several years. We reached the conclusion that DR2518, which we are referring now here in the entire story, was characterized as a radiation responsive serine pyramid type quinoprotein kinase. We name it as RQKA. Now, absolutely, I have brought to the conclusion of my slide. I don't want to load you with lot of biological jargons, but eventually, this is a summary slide. What all research has been done, starting from a chemical molecule which was identified in this bacterium and leading to a discovery of a novel pathway which has never been spoken in the scientific literature before that. So what it says, 25 years research actually, that gamma radiation, that the DNA damage produce the ROS, all these things I am talking about. And then you see the induction of the RQK, that is the DR25181 component. You see the induced synthesis of PQQA, that is an antioxidant and prooxidant, which we were talking. PQQ actually can regulate the activity of this enzyme. You know, PQQ is a chemical molecule. Enzyme is a protein macromolecule, which Tanvir gave some introduction to you. But this actually work together, and the activity of this protein can be activated by PQQ. Now this kinase actually phosphorylates whole lot of repertoire of proteins, macromolecules in the cells. Some of them they start doing the DNA repair and the other will arrest the cell division. Now this particular thing pay the attention because whenever cells are exposed in any biological system to gamma radiation, DNA is damaged, then two processes will have which are bound to happen. Number one, that the damaged DNA will have to be repaired if organism is going to survive. On the other hand, the cell division should not continue. Cell division should stop. And this kind of thing actually we reached to a discovery kind of science where we demonstrated that phosphorylation by RQK protein actually can regulate both the process. It can induce the DNA repair on one hand and it can arrest the cell division on the other hand. Now what had been known actually in the previous literature, there was a different mechanism was discovered for doing this job in bacteria. So far, nobody has talked about DNA damage response and cell cycle regulation. Actually, that how DNA repair is increased, how cell division actually arrested, other than this mechanism, which is called, let's say, Recce type canonical SOS response. This is something has been in the literature for last 70 years in bacterial research. But from our work, what we demonstrated for last 25 years, it actually concluded that our study provides a possible alternate mechanism to, let's say, Recce type canonical DNA damage and cell cycle regulation in bacteria. And bacteria may have cell cycle regulation similar to eukaryotic system, that is human or plant or higher organism. So this is something, if I want to conclude it at this here, suggesting that every our cells in the life form in the living system, in the human, plants, yeast, bacteria, fish, molluscan, all have got a lot of storage of chemical compounds, and these compounds they are actually important both in the drug discovery, something antioxidants, discovery of molecules, but the basic mechanism eventually happens by controlling the function of the macromolecules, that is metabolic processes. I come to the end of it. I hope I have concluded. This is a popular group which I have, very beautiful people who have worked for several years with me directly before I became the head of a big division in Naga Atomic Research Center. This was a small my working research group, which we have research group and then we also have administration job to do. The entire work that I have presented of Dr. Nivedita Khairnar, actually she started the work, continued by T. Mehta, Modi Mehta, and then we have Dr. Y.S. Raj Purohit. He has actually made all the contribution in the work that I have presented. And subsequently, Dr. Ganesh Maurya, he is the faculty after doing PhD in PHU. And he did, and then subsequently, the work is being picked up by younger people who are in the front row. They all have been my PhD students. Now they have become officers in Baba Atomic Research Center after all these things, and I'm grateful to all of them. Of course, DRC, Department of Atomic Energy, which have been my venue for bread butter. Many more. This is a very important message. The life, whenever we want to do research or go in career, it's never rosy. It's always had a hurdle, but I think we should always acknowledge to those who have kept testing your perseverance in your career. And with this, I would like to thank you for having the patience hearing. I have exceeded some time, but bear with me. This is something which I am still sitting excited that we'll do a lot of research in future. Thank you so much. There are questions I would be happy to answer. Hello.
Thank you, Professor Mishra. Now,
Yeah, if there are questions, I would be happy to answer it.
perhaps I don't know time is permitting or not. Should I come there?
Yeah, hello. Go ahead. Yeah, please. I think. Yeah, go
Thank,
ahead. Yes, sir.
thank you, sir. Thank you, Dr. Misra, for giving a very valuable and pertinent topic in the front of chemists, biologists, and other people. Although you're such an informative lecture, give a pandera of questions, but since you have a lack of time, I request the audience, if they have any question, they can submit to the organizing committee of the conference, or they can also send to the Dr. Misra, and I hope Dr. Misra will respond their questions as per your convenience and as per the satisfaction. The first lecture delivered by Dr. Tanveer Khan. He has also delivered a very nice talk on the medicinal chemistry, and the questions also already organizer has requested. If you have any question, you can submit to the organizing team. As well, you can respond them immediately. So with these words, I conclude this technical session and hand over this podium to the organizing committee. And thanks for giving me opportunity to chair this session. Thank you, sir.
Thank you. Thank you, everyone.
Thank you, Professor Misra sir, for accepting our request to present in the conference. Thank you once again for sharing your prestigious and enlightening research to us. Our students and young faculty members of the conference will be benefited from your talk. Thank you, sir. Thank you.
Thank you. Thank you, everyone. See you. Now it's a good morning for me and good evening for you.
Thank you, sir. Now we have the third talk of the first technical session of day second. The speaker of this talk is Professor Muhammad Adnan Khan. The chair will be the same one. I'll read out his biodata. At the very outset, I would like to say that his father, Muhammad Adnan Khan's father, was an alumni of this college. He, he did his MSc from King Abdullah University of Science and Technology, Kingdom of Saudi Arabia, in Material Science and Engineering. Did PhD from the same university, King Abdullah University of Science and Technology, in the same branch, Material Engineering, and he has experience of 10 years in leading research, industry, and academia. He has published approximately 41 research journals in international reputed journals. He has, to his credit, 4 industry, government reports, 6 granted US patents. Currently, he is working as Assistant Professor, Department of Chemical and Material Engineering, University of Alberta, Canada. His research interest is development of photoelectrocatalysts, reactor systems for renewable hydrogen and chemical production, development of integrated energy systems models based on techno-economic and life cycle analysis to help build credible net zero transition pathways with a focus on role of hydrogen. The topic of today's talk is hydrogen and the decarbonization of steel production in Canada. Dr. Adnan, over to you now.
Is my screen visible?
You are visible. Your slide is also visible. You are also visible.
Okay, all right. Thank you so much for the kind introduction. Good afternoon, everyone. All the faculty members, all the young students attending the conference. First of all, let me say it's such a pleasure to be here and have the opportunity to present my work, and I thank the organizing committee for the invitation. So my name is Mohammad Adnan Khan. I'm an assistant professor at the University of Alberta. We are situated in Edmonton, which is a city in Alberta, Canada. And today the title of my talk is Hydrogen and the Decarbonization of Steel Production in Canada. Now, before I begin my talk today, there's a couple of statements I need to make. The first one is, you will probably see that my talk is of a very different flavor to all the different talks in the conference. So it's very applied in that sense, and it is basically considers a systems level approach on how to decarbonize the energy sector. This is a very important research direction for engineers and young students to really develop net zero solutions in the future. The second statement I want to make, while the study itself is focused on the steel production in Canada, the approach and methodology used here is applicable for India and jurisdictions around the world. All right. Before I dive into my talk today, a quick snapshot of my research group at the University of Alberta. We call the Energy Transition Lab. Basically, have two arms of the research. I'll just point out there's a pointer. We have the experimental front of the research, which focuses on developing catalysts, and this is all fundamental science regarding development of electrocatalysts for renewable fuels and chemical production. A lot of the focus is on biomass oxidation and methane oxidation to produce renewable fuels and chemicals. The other part of the research group is really about developing energy systems model, primarily using techno-economic analysis and life cycle analysis to develop credible net zero transition pathways. And like highlighted in the introduction, a lot of my interest is really around the role of hydrogen for different energy sectors, right? And that is what I'm going to focus on, is really the role of hydrogen for steel production. So my presentation today has 4 different sections. The first section is called steel production routes, energy use and emissions. Then I'll basically go to the core of the study, which was a case study around the adoption of hydrogen for steel production in the Hamilton region. Hamilton is a region where we produce steel in Canada. And the steel industry, how the steel industry can act as an anchor tenant for a larger hydrogen economy, and some key recommendations. Like I said, the study was framed around steel production in Canada, but the approach, methodology, and results is also very relevant for India. Okay. So energy use and emissions associated with Canadian steel production. So the slide I have here is about steel making in Canada. Just added a few facts and figures about India as well. Now, regarding Canada, we are among the top 3 producers of steel in the world. We produce about 13 billion tons of steel. But this dwarfs against what India produces. India is the second largest steel producer in the world, producing about 154 million tons of steel. And steel is an essential product in the entire net zero transition. Steel is deep-rooted in our society and used in every aspect of our life. And when we talk about CO2 emissions from steel making in Canada, there is about 15 million tons of CO2 emissions from steel production. India is about 297 million tons of CO2 emissions. That's significant. There's a fact I read, and which needs to be double-checked, that these emissions from steel production in India are about 12% of the country's total CO2 emissions, which is very significant. So this is a very important sector that India needs to focus on decarbonizing. And when I talk about steel production, the blast furnace or the blast oxygen furnace route has the largest share of primary steel production. I will go through what this route means. But in the past, the Canadian steel industry has heavily invested in reducing energy consumption and emissions from this sector. And this is highlighted in the emission intensity graph here, where you can see Canada has the lowest emission intensity for primary steel production, while India is among the top three highest. So India is right here, and Canada is here. So there's a long way to go for India to really look at decarbonizing this industry. Now, when I talk about primary steel production, the slide I have here covers three primary steel making routes. The blast furnace, followed by the blast oxygen furnace route, which basically uses coal or coke as a reducing agent and also as an energy source. For reducing iron ore in the blast furnace, you basically convert the coal into carbon monoxide, which is a reducing agent, and then you make a pig iron, which is further reduced or further purified in a blast oxygen furnace to make your crude steel. Other alternative routes to making steel is the DRI route, which is direct reduced iron. So typically this uses natural gas as a reducing agent and also produces energy use. So you have shaft furnace, and the key aspect of the direct reduced iron is basically you reduce iron ore below its melting point in its solid state, right? And you make direct reduced iron, which is further purified in an electric arc furnace. The focus of this study is around hydrogen DRI. So this is identical to the natural gas DRI route, but instead of using natural gas, we use hydrogen as a reducing agent, but also for providing the energy required for the process, right? And the important fact to highlight here is a couple of important facts actually. The hydrogen used in this process will only make sense if it comes from low carbon sources. So either from water electrolysis powered by renewable electricity or via blue hydrogen, which is basically steam methane reforming coupled with carbon capture and storage. Right? The second important point, hydrogen DRI is a little tricky because it's an endothermic process. So you need extra thermal energy to maintain the shaft temperature, and that I will highlight in a couple of slides as well. So I have here a breakdown of energy use and emissions with primary steel production, all the three different routes. The left-hand side is basically all results of this study, and this is from another source in literature. What you see is 2 points, and I'll highlight this very clearly. The DRI routes are more energy efficient than the traditional blast furnace blast oxygen furnace route. Right? It can be fueled with either natural gas or hydrogen, like I said. The impact of DRI, so compared to blast furnace blast oxygen furnace, there is significant emission reduction, scope 1 emissions, with the use of the DRI process. With the use of natural gas, you can reduce it about 60 to 70%. With the use of hydrogen, you can almost reduce the scope of emissions to nearly zero. So significant advantage of transitioning to hydrogen DRI. But apart from these advantages, there are also challenges with transitioning to hydrogen DRI. What are the challenges? The main challenge is the cost of energy. So assuming a natural gas $5.50 a gigajoule, let's without carbon taxes, in today's scenario, best case scenario, hydrogen price, let's assume 3 to 5, I'm talking Canadian dollars a kg of hydrogen, we're talking about anywhere between $20 to $35 a gigajoule. So you're really looking at the price going 4 to 5 times higher when transitioning from natural gas DRI to hydrogen DRI. So the key message I want to highlight at the end of this slide is minimizing the cost of hydrogen supply will be critical if we really have to make hydrogen DRI work. So how do we really do that? When I talk about reducing the cost of hydrogen delivered for steel industry, let's talk about production first. So like I said, you can make low carbon hydrogen via electrolysis. In today's scenario, based on the current cost of electrolyzers, when you can produce hydrogen, produce hydrogen via water electrolysis, but the cost of that hydrogen will be quite expensive, right? So I have on the graph here is levelized cost of hydrogen production as a function of electrolyzer use and the cost of electricity. The cost of the electricity is the key determinant which dictates the cost of the hydrogen. In today's scenario, you need very, very cheap electricity at 3 cents a kilowatt hour or 30 cents or $30 a megawatt hour to produce low cost hydrogen at $3 a kg of hydrogen. And this has to be available with high capacity factors. Right? Even in the future, with electrolyzer prices going down, we still will be significantly dependent on the electricity price, right, to dictate the cost of the hydrogen production. versus natural gas reforming, a mature technology, where most of the hydrogen in today's scenario is what made by steam methane reforming. And when coupled to carbon capture and storage, you can make low carbon hydrogen via natural gas reforming. This process, the cost of the hydrogen produced, significantly depends on the natural gas price. But the most important point to highlight here is, irrespective of the natural gas price, even if you go up to 6 or 8 Canadian dollars a kg, you can still make hydrogen at very low cost, about 2 to 2.5 Canadian dollars a kg of hydrogen, right? Obviously, the key point to highlight here is you require the right geology for permanent CO2 storage. All right. So this is really setting the context, but let's look at a particular case study about the adoption of hydrogen in the Hamilton region. So in Canada, there are 3 primary steel making plants, which is all located in southern Ontario. So this is a map of Canada, this is United States. These 3 red dots are our 3 primary steel making facilities. In this particular study, we basically were looking at the ArcelorMittal Dofasco plant, which is right here, if you can see my cursor. And this is an integrated steel mill that has already announced plans to transition to a DRI facility, but that DRI facility will use natural gas, but will be ready to use 100% pure hydrogen in the future. Right? So let's look at this particular plant. Right? And this is that systems level approach that I was highlighting at the start of my presentation about how do you transition entire sectors and how do you develop credible net zero solutions for different sectors. So if you talk about the Dofasco site, this is the 2.5 million ton DRI capacity, right? The estimated hydrogen demand for this particular plant would be about 492 tons of hydrogen per day. Just one single steel plant will require almost 500 tons of hydrogen per day. This is 6% of the current hydrogen we produce in Canada, right? And let's suppose I want to produce this via water electrolysis, right? There are really big supporters of water electrolysis, but sometimes we need to think about credible solutions. To produce this much hydrogen via water electrolysis, we will require 27 gigawatt hours of electricity. That is 6.5% of the entire province's power generation. It's almost half a nuclear power plant, right? So that's really not feasible, right? If electricity has to be delivered to this, and again, I talked about the cost in the previous slide, you're really looking at very expensive hydrogen for water electrolysis. So the question that comes to mind, it is difficult to see how green hydrogen produced at the Dofasco site could be a cost-effective solution. So what about blue hydrogen, right? That's the second question that comes to your mind. And when I talk about blue hydrogen, like I already said previously, you need CO2 sequestration capability, right? And how much CO2 sequestration do you need? At 500 tons or 492 tons of hydrogen per day, you need about 1.6 million tons of CO2 per year. So if you look at Canada, what we identified is potential CO2 storage sites. All of the neighboring CO2 storage we have in Canada is actually across the border in the United States. Right here in the provinces of, the states of Ohio, Pennsylvania, Michigan, but the potential is huge. This is the identified potential, CO2 storage potential in these 3 provinces is enough to store hydrogen from this single plant for 38,000 years. So there's no shortage of CO2 storage potential, right? So what we came up with or looked at this in study is the potential of actually making hydrogen in these provinces in the United States and then pipelining the hydrogen 400 kilometers to the steel plant in Canada, right? And the advantage of doing this is because these regions have very low cost natural gas, but also have the right sites for CO2 sequestration. So when you look at this, then you're able to identify credible solutions of how can I deliver hydrogen, at what cost can I deliver hydrogen, and whether it makes economic and environmental sense for the sector, right? So what we did is develop hydrogen pipeline models, and we do the gas flow calculations and do the detailed techno-economic models, and we look at hydrogen pipelining costs. At these scales, and we've done at different scales, 246 to 492 tons of hydrogen per day, the cost of delivering hydrogen via pipelines will vary anything between about 1 to 1.6 Canadian dollars a kg of hydrogen, right? And if you add the production cost, the hydrogen compression cost, and CO2 this cost, you get delivered hydrogen cost, which is anywhere between 3 to 4 Canadian dollars, depending on the scales of delivered hydrogen. And that basically tells me that at 3 Canadian dollars, so at 492 tons, best case scenario, I'm 3 Canadian dollars. At 3 Canadian dollars, I'm still at $21 a gigajoule, which is still about 4 times more expensive than a natural gas DRI process. The energy cost will be 4 times more expensive. So the next thing, how do you really make this work? The problems with a dedicated pipeline for a single plant is risky, right? There is still a high price for energy supply. There is a higher risk and less likely to attract public support and investors, right? Whenever you think about a systems level approach, you need to think about energy different sectors and how different economic sectors are linked and interdependent on each other, right? And that's the next question we try to answer. Are there potential markets for low carbon hydrogen that could help reduce this cost for the steel industry? And that's where we looked at, and what we developed is called macroeconomic models to look at potential hydrogen demand in the province in that region, right? For example, I will just show you one particular sector and how we developed these models. This was replicated for different sectors. So transportation is a key one. So transportation in Ontario 2019, we used about 943 petajoules of energy to power transportation in the province. And this is divided into sectors such as light duty vehicles, medium duty vehicles, heavy duty trucks. And the first question is, what are the most critical net zero solutions for these sectors? On top of it, on top of the inner circle and the inner pie chart, what I've identified here is possible net zero options for displacing current fossil fuels. Current fossil fuels, you basically use diesel and gasoline transportation. In a net zero future, we're talking about powering that sector via electricity or hydrogen or a small amount of biofuels. We only have 3 credible net zero fuels that we're really talking about for transportation, right? The majority of transportation will go towards electrification, but heavy duty transport is a sector that could be a very attractive sector for using low carbon hydrogen. The outer circle, what you see here, is a possible improvement, efficiency improvement, because all of these net zero options are much more efficient than fossil fuel based combustion engines. So battery electric vehicles are much more efficient. Hydrogen fuel electric vehicles are more efficient than diesel engines. So this will lead to efficiency improvement of the entire sector. And this is reflected by the outer screen. And we did this for other sectors I was highlighting here. For example, agriculture here, and will not go through the calculations, but the concept is the same. And when you put together the numbers, what you realize is, okay, this is very interesting. Look at transportation. We need an additional 33 terawatt hours of electricity, but we need an additional demand of 3,607 tons of hydrogen per day. Okay? And then we started doing this for all the different sectors in the province: transportation, agriculture, residential and commercial buildings. And what we come up with is macro models, and these are used with the government of Canada to really develop transition strategies. And what we realized for the province of Ontario, if based on the 2019 energy, if you have to transition to a net zero future, which is on the right hand side, you have to double your electricity production. You have to double your public electricity grid. You have to more than double your biomass production. Your CCS storage has to be 3.3 million tons of CO2 at least. Right? But the most important number I want to highlight here is the demand for low carbon hydrogen with about 12,000 tons of hydrogen per day. It's huge. Because today we barely use any hydrogen. We use hydrogen in the chemical sector, right? Not as a fuel. So truly to drive to net zero, this is the demand of hydrogen you're talking about. And when you look at that demand, and I'll try and wrap it up in the next couple of slides. So earlier I told you about a single plant, but what about I can supply hydrogen not only to the steel sector, but also across multiple sectors together. If I do that, I will reduce the cost of pipelining hydrogen by more than 50%. Okay? And when I add on top of that the delivered cost of hydrogen, what I realized is we can really deliver hydrogen at 2.5 Canadian dollars a kg of hydrogen. And at 2.5 Canadian dollars a kg hydrogen, we slowly start to make hydrogen DRI very attractive as an economically feasible route for primary steel production. And what does 2.5 for the steel sector mean? And this is my last slide here is, if you remember one of the first slides I showed you energy demand calculations, GHG emission calculations for the different provinces, and I highlighted the transition from natural gas DRI process to hydrogen DRI process will lead to reduced scope 1 emissions from 333 to 555 kgs of CO2 per ton of steel. That's the rough range. But it will also increase fuel cost by about 112 to 121 Canadian dollars per ton of steel, right? And this is at 2.5 dollars a kg of hydrogen. What does this really mean? At this cost, how do you really make it work? If you divide both of these numbers, what you really understand is the cost of CO2 abatement. The cost of CO2 abatement by transitioning to hydrogen DRI will be in the range of $200 to $360 a ton of CO2. Still very high, but not impossible anymore. Because in Canada today, we're paying about $70 a ton of CO2, right? In 2030, the federal government mandate is about $170 per ton of CO2 emitted. So that's not very far from $200, right? So really starts to make sense. And if I put all of this together, I can put together a key recommendation for the entire segment. The first one would be, it is very important to reinvest revenue from carbon pricing to decarbonize steel making. It is very important to reach economies of scale and work with multiple sectors and the province and the government together to deploy a regional strategy around decarbonization and around the adoption of hydrogen. It's also, for us in Canada, it's also very important to work together with our neighbors down south in the United States and understand the impact of different policies on the low carbon hydrogen production. And also, it's very important to monitor the progress of new innovative technologies such as turquoise hydrogen, which is basically methane pyrolysis. And also, it's very important for the government to work together to build up demand for low GHG steel. And like I said, all of these recommendations are very applicable for India as well. It is very important to sit together, government together with industry players, to really develop these transition models for different sectors, in particular the steel sector. My final slide, I just want to acknowledge everyone involved in the study, the funding from the federal government of Canada via the Natural Resources Canada, the steering committee, which is the Canadian Steel Producers Association. We're collaborating with other authors from a non-profit organization such as the Transition Accelerator and Thorn Associates. I'd like to acknowledge everyone involved in the study, all the co-authors and the students involved. Thank you so much. We have big questions right now. Send me your questions, please. Thank you so much.
Thank you so much, Adnan sir. Thank you so much. हाँ सर, वहीं से बोलिए सर.
Thank you, Dr. Khan, for delivering such a nice lecture pertaining to a very important field of science. I feel there are a lot of questions with the audience, but perhaps the time is an important factor which doesn't allow to interact you. And I request you, if the organizing committee is sharing their questions, you will provide the answer to the audience. With these words, I once again thank you for gracing this opportunity. Thank you, sir.
Thank you very much. Thank you, everyone.
It's okay. I hope we meet you soon, sir. Now our next talk is given by Dr. Vikram Singh. I want to introduce him in very short. Dr. Vikram Singh has completed his PG and PhD, PG from University of Lucknow in 2010. and he done his PhD from IIT Madras in 2016. He completed his postdoc from CDRI, National Postdoctoral Fellowship, from 2016 to 2018, and another postdoc, Institute Postdoctoral Fellowship from IIT Kanpur from 2018 to 2021. He has published more than 22 research papers in reputed journals, and he filed 2 patents. He awarded Madhya Young Technological Innovations Award at Rashtrapati Bhavan in 2017, and for his PhD thesis, he got Langmore Best PhD Thesis Award in Physical Chemistry in IIT Madras in 2016. Currently, he is working as a scientist and assistant professor in Central Institute of Mining and Fuel Research, Dhanbad. The topic of today's talk is development of white light emitting materials. Now, Dr. Vikram Singh, start your lecture.
Hello. So, good afternoon to all of you. First of all, I would say thanks to organizer for providing this opportunity to deliver my talk here, especially Dr. Arun Kumar. And the topic of my presentation is development of white light emitting materials. So first, I would like to tell about what is the white light emission. As we all are aware that sunlight is composed of 7 colors. That is, sunlight is composed of 7 colors. That is violet, indigo, blue, green, yellow, orange, and red. When the sunlight refracted to in the face, it gives all 7 colors. But idea in natural white light emission, what we are using in LEDs, tunable lasers, that can be used for the composing of red, green, and blue. When we mix these 3 colors in an equal ratio, that is 33% red, 33% green, and 33% of blue, then we can get perfect white light emission. Next. So generally white light emission typically involves the mixing of 3 primary colors that I told you in the previous slides. Red, green, and blue are combining of 2 complementary colors. Means complementary colors means when you are mixing blue and green, you can get cyan color, which is coming around 475 nanometers. So if you are taking cyan color and this is the complementary color, and you are taking another primary color that is a red, then by mixing of complementary color and primary colors, you can get the white light emission. These are the possible applications of white light emitting materials in the field of LEDs lamp, because LEDs are mainly fabricated by the rare earth elements, are critical elements like indium, gallium, and these rare earth elements, we don't have sources in our country. We are totally dependent on the China, which has exporting the rare earth elements for the LEDs fabrication, and these are the very costly. Also, we can explore these materials in the tunable dye lasers. Tunable dye lasers means by changing the concentration of one of the emitting pigments or one of the emitting colors, we can tune this. We can explore this material in the tunable laser, flat panel display, and backlights. Next. Before going into detail, first I would like to introduce about how we can predict that this material gives the perfect white light. By calculating the CIE color temperature, CIE color coordinate and color temperature, we can predict that this material gives the perfect white light emission. When we are getting the X and Y, that is the chromaticity index, that is if it is coming about 0.33 and 0.33, then we can say this is the perfect white light emitting material. And from this color coordinate, we can calculate the color temperature. Every emitting sources have its color temperature. Nowadays, in the restaurants, people are changing the lights, LEDs lights. If they are using the white LEDs, warm LEDs, cool LEDs, these warm, cool, and cool LEDs based on their color temperature. So from they are tuning this correlated color temperature, and color temperature around 500 Kelvin, that is the cool color. And if the color temperature is coming around 2700 to 3000 Kelvin, that is warm color. Next. So this is the correlated color temperature. When if the correlated color temperature is coming around 7000 to 10,000 Kelvin, it is called daylight or natural color. And below to that, 4000 to 6000 Kelvin, that is the cool white light, and 1000 to 3000, that is the warm white light color temperature. Next. So in the literature, people are choosing the different types of material like inorganic, organic, nanomaterials, quantum dots, polymer, metal organic framework, inorganic or organic hybrids, metal complexes, and lanthanide doped material for the preparation of white light emitting materials. By applying the various photophysical principles, they are getting the white light emitting material by employing hydrogen bonding, mediated J aggregation, mixing of monomer and hexamer solution. But in our approach is totally different what people have done earlier. That is, we are focusing on the preparation of fluorescent carbon-based nanomaterials for white light emission using a cost-effective and environmental benign approach. Next. So why we have chosen the carbon nanomaterials? Because carbon nanomaterials are very cost, we can prepare these materials in a very cost effective and environmental benign process. They have very small size in the range of 1 to 10 nanometer, and these nanomaterials can be prepared by using two techniques like bottom up and top down approaches, applying various different types of synthesis approaches. Next. This is the possible application of fluorescent carbon nanomaterials over the metal-based quantum dots and organic dyes like excitation-dependent emission. As we see, as we change the excitation wavelength 450 to 450 to when we are changing the excitation wavelength, there is change in the fluorescence spectra, emission intensity. This is the very important phenomena. But organic dyes emits, if whatever the excitation wavelength, organic dyes have a fixed emission. But in case of nanomaterials, we can change its the fluorescence properties. And high water solubility, excellent photostability, easy surface functionalization, good biocompatibility, and low toxicity. These are the multifunctional These are the multifunction application of fluorescent carbon quantum dots in the field of drug delivery, optronics, catalysis, sensors, theranostic, and bioimaging. Next. So this is we have prepared white light emitting graphene quantum dots from coal soot, and we have filed Indian patent for that. We have, in the literature, most of the report examples which achieved white light emission by mixing of red, green, and blue. As I told you previously, are complementary color. Then the first time we have prepared white light emitting graphene quantum dot from coal soot from a single source and single material. We did not use red, green, and blue for mixing, and the developed process aids in the value addition and gain full utilization of coal soot based for the production of white light emitting material. So we have taken the 1 milligram of coal soot, and this coal soot we have collected in our department from anthracite coal, and then we have burned this coal and collected the coal soot in the glass plates, and then after this coal soot, we have mixed this coal soot with ethylene diamine of 10 ml and ultrasonicated at 2 minutes for room temperature. Then we have get the reaction mixture of ethylene diamine, functionalized this graphene quantum dots, and then we have centrifuged this reaction mixture at 7000 RPM for 10 minutes. We have found the graphene quantum dots. This is the visible light. In the normal light, we have obtained the yellowish color of the graphene quantum dots. Next. We have not only focused on the ethylene diamine, we have explored this graphene quantum cold suit in the various solvents like H2O, ethanol, THF, DMF, DMSO, and ethylene diamine. But we did not see any white light emission in the case of H2O, ethanol, and THF because of in this solvent we are getting only the monomer emission, which is coming from the blue region. You can see here we are getting a single peak at near about 450 or 475, which is a blue fluorescent materials. But in case of ethylene diamine, we are getting two bands. One band is coming about 475 nanometer, and one SAR band is coming around 625 nanometer. So this is the mixture of two colors. One is complementary color, and this is the red color. So this in the case of ethylene diamine, we can see the emission spectra is cover all the visible region from 400 to almost 700 nanometers, which that is why we are getting the white light emission in the case of ethylene diamine. Because in the case of ethylene diamine, we are getting the white light, we are getting the J aggregation, and so one peak is coming from the monomer and another peak is coming from the J aggregation. That is why and we have calculated the CIE color coordinate, which is coming around 0.28 and 0.33, which is close to the perfect white light emitting coordinates. And the color temperature we have calculated 8,479 Kelvin. We have calculated the size and morphology of prepared graphene quantum dots, and we have found that this graphene quantum dots has just 8 nanometer of nano size and have the graphitic nature, which is from HR10 we can see there is a spacing. We have found the 0.35 nanometer, and their graphitic nature was also confirmed by the XRD and Raman. We have found two bands, that is the graphitic bands and disordered bands in the case of Raman spectra, which confirms the graphitic nature of the graphene quantum dots. And XRD, which is from the XRD spectra at the 2 theta, we have found a broad peak 23.8, which corresponds to the interlayer spacing of 0.35, which is correlated with the HR10 analysis. From HR10, we have calculated the interplanar spacing, that is 0.35 nanometer, and from the HRD, from XRD analysis, we have also calculated the interplanar spacing 0.35 nanometer at 2 theta is 23.8. Next. We have also did the functionalization, detailed XPS study to get the clear picture of the graphene quantum dots, and we have found that this graphene quantum dot is composed of carbon, of nitrogen, and oxygen, and they are well functionalized with ammonium cation and carbonyl group, carboxylic groups. Next. Then this white light emitting materials we have mixed with this white light emitting solution with PVA film, polymer matrix. So we have found the perfect white light emission in the after mixing this this graphene quantum dot in PVA solution, and we have found perfect white light emitting under UV light excitation, and we have found the quantum yield is 0.52, which is good enough to the compare with the other reported methods, and CI value is 0.28 and 0.33. So the white light emitting material is only useful in the solid state. If you are achieving the white light emission in solution, it is not useful for the technology development. So we have used this material, and we have tried to mix this material in the polymer matrix, which gives the perfect white light emission. Next. So we have also seen that by changing the graphene quantum dot concentration, there is shift from blue to red, that is the J aggregation. See, as at when the concentration of graphene quantum dot is 21 microgram per ml, we have found the perfect white light coordinates, which is 0.28 and 0.33 with color coordinate 8,479 Kelvin. When the concentration of this white light emitting white light graphene quantum dots changes from 1.3 microgram per ml to 168 microgram per ml, there is shift in color coordinate from white to red because of J aggregation. Next. This is the possible mechanism of J aggregation. This is the the synthetic representation of graphene sheets, which is functionalized with the ammonium cation, and these ammonium cations have tendency to form electrostatic interaction with carboxylate ion and hydrogen bonding. So these two phenomena is also suggest that this is the J aggregation formation. Because of J aggregation formation only happen in the case when the molecule have electrostatic and hydrogen bonding interactions. Next. Next. These J aggregation studies have been further verified by the lifetime analysis, and we have found that the fluorescence lifetime of monomer is greater than the lifetime decay of J aggregate. So we have confirmed that proposed hypothesis by fluorescence lifetime and zeta potential measurement, because the zeta potential is also coming around 0, so which suggests that there is a formation of aggregates. There is no positive or negative charge are present in the graphene quantum dots because of the electrostatic and hydrogen bonding interaction. The zeta potential is coming almost 0. Next. We have used this white light emitting materials. So this is the UV LEDs, which is coming at 380 nanometer. Then we have coated this UV LED with PVA matrix, graphene quantum dots, and by on applying the voltage of 3 volt, we can see the ultraviolet LEDs gives the white light emission, and we were able to distinguish all the colors. We can distinguish any colors only possible by the white light emission. If you want to distinguish all the colors in the presence of red color, not possible. Green, not possible. So you can able to distinguish red, green, blue, yellow, whatever in the case of white light emission. So here we can see that on illumination of white light emitting graphene quantum dot after coating in the UV LEDs, we were able to distinguish the yellow, blue, green, and red color. Next. In the second line of work, and this is my PhD work, we have also prepared the white light emission from the mixture of pomegranate extract and its carbon nanoparticles, and this is published in Journal of Material Chemistry C. So we have prepared the green emitting carbon nanomaterials from pomegranate extract by doing the hydrothermal treatment at 140 degrees centigrade for 24 hours, and then centrifuge. We have found the yellow emitting carbon nanomaterials, which gives the 11 nanometer of size and have carbon and oxygen elemental composition in its surface with carboxylic acid functional group functionalities in its surface. So we have prepared the green emitting carbon nanomaterials from pomegranate extract, and we have characterized using the HRTEM and XPS study. Next. We have also done the detailed optical properties of carbon nanoparticles and pomegranate extract. We have found that this pomegranate extract gives the 2 band, which is coming around blue region. It's because of ellagic acid. Pomegranate juice has ellagic acid and anthocyanins, this compound. So the blue fluorescent is coming around, which is coming around 444 nanometer. In the blue region, it's because of ellagic acid, and this is coming around 595 nanometers because of anthocyanin. So we thought we are getting the blue and red in our materials. So we thought by mixing the green fluorescence, green emitting material, we can get the perfect white light emitting material for which covers the entire visible regions. And we have calculated the color coordinates of both the compounds. That is green emitting carbon nanomaterial, which is coming around 0.26 and 0.41, whereas for pomegranate extract gives the 0.23 and 0.20. So we thought by mixing these two components, we can get the white light emitting materials. Next. So we start mixing these. We start mixing the pomegranate. This is the carbon nanomaterials in the pomegranate extract, and we have found the perfect color coordinate at this is at the J point. This is the point 0.30 and 0.33, and this is not a simple mixing. If it is a simple mixing, we can get a straight line in the chromaticity diagram. But we are getting a trajectory in the color coordinate diagram. It's because of the FRET, because of there is a spectral overlap between the ultraviolet of UV visible spectrum of pomegranate extract with the fluorescence of carbon nanoparticles. So there is a chance of energy transfer uh, from from carbon nanoparticle to pomegranate extract. That is why the intensity coming around 595 nanometer is increases as we change, as we are adding the carbon nanoparticle concentration from let's say 0.15 mg per ml, 3 mg per ml, 0.3 mg per ml. So there is significant enhancement at the at the emission at 595 nanometers because of the energy transfer from carbon nanoparticle to pomegranate extract. So we have optimized the concentration of pomegranate extract and carbon nanoparticle in white light emitting materials, and which is coming around 2.65 to 26 micromolar, and the concentration of carbon nanoparticles coming around 0.3 mg per ml, and we have achieved this white light emission in solution as well as in polymeric film. So in conclusion, we have utilize the gainful utilization of coal soot for the production of white light emitting materials, white light emitting graphene quantum dot. That is the best to build approach. And we have another line of work. We have also achieved the white light emitting materials from single plant component, that is the single fruit component, that is a pomegranate extract. After doing the hydrothermal treatment, we were able to achieve the fluorescent carbon nanoparticle which gives the green emission. And after mixing this green emission with pomegranate extract, by utilizing the Foster resonance energy transfer, we were able to get the white light emitting material which covers the entire visible region. So this is the couple of work we have done in the field of optoelectronic application of white light carbon nanomaterials. Next. So with this all, thank you. Thank you so much to all of you for your kind attention.
Thank you. Thank you, Dr. Singh. Now, we can invite a couple of questions, preferably from the student section, because the lecture deals with the very common topics, waste to wealth conversion, and very popular application. Please, sir.
Right. See, these are the colloidal solutions. So these are the colloidal solutions. They are dispersed in the medium. They can be dispersed in the medium. So you can isolate by the centrifugation technique, but you're not possible for the quantum dot.
मेडिकल एप्लीकेशन में लेकर जाना है और जिस टाइम सपोज वी आर सिंथेसाइजिंग क्वांटम डॉट्स देन अदर एडिटिव्स वी आर एडिंग so if suppose that solution is being used for suppose killing cancer cell or something like that, so how to establish that the cancer cell is getting killed by quantum dot or the metal ions present in the solution if they are not separated from the solution?
You are when you are preparing the quantum dots, there is a purification steps. So during the purification steps, we are using the 1 kilodalton of dialysis membrane.
Yes.
So in that purification steps, so most of the heavy metals or other metals are present, so they are coming out from the membrane.
So only dialysis membrane is the way, because it takes
the better way to.
It takes too much time using dialysis membrane.
Yeah, it takes more than 24 hours.
Yes.
Any
Thank
student? Young student? One of our colleague want to ask one question.
Thank you, Dr. Singh. Just there is a query in slide number 14. You said that after preparing your this GQD, that you get it in a form of emulsion. Then you have to, for the application part, you have to use it on some polymer to get it in a solidified phase. So what polymer have you used for that?
PVA.
PVA.
Polyvinyl alcohol.
So, if you don't have any question, although invite many questions, but once again I thanks the Dr. Singh for delivering a nice question. Thank you.
Thank you, Dr. Sir. Now, this session, the technical session ends here. I request the chairpersons there, they should take their seat in the auditorium. Now, next session will start. Before I speak something else, I would like to say that whosoever is present in the auditorium right now, they will take lunch. Please don't go before taking lunch. हाँ, जिनके घर पे अभी बना भी नहीं होगा हाँ अब वो हो गया, देखिए वहीं से हो गया है नहीं, it is actually live, हाँ, it is on YouTube now. Dr. Farke, it is live actually. Okay. So we are going to start the second technical session of day 2. For this session, I would like to invite Dr. Khem Bahadur Thapa, Associate Professor, Baba Saheb Bhim Rao Ambedkar Central University, Lucknow, as chairperson of this session. Please take your seat. Dr. Manish Gupta, JP University, Chhapra, as co-chairperson. Please take your seat. Dr. Manish Gupta. Is he here? Dr. Manish Gupta? He is here actually. Dr. Manish Gupta. He will act as co-chairperson of this technical session. Dr. Muhammad Akmal. He will be reporter of this session. Please take your seat. The speaker is Professor P. Gopalan, Professor P. Venu Gopalan, sorry. To introduce him, I would like to call upon Dr. J.K. Pandey, Assistant Professor, M.L.K, Dr. J.K. Bhatt, Dr. Jiten Kumar Bhatt, Assistant Professor, M.L.K PG College, Balrampur.
Thank you, sir. And thank you everyone for coming in this conference on the second day. I am Dr. Jiten Kumar and I have been given the responsibility of introducing one of the best person I have met in my life. When I was struggling after my PhD and postdoc, I received the Young Scientist Award from DST, 40 lakh funding I received, and I joined Professor P. Venugopalan's lab in Punjab University. At that time, he was the chairperson of the department. So today I have this opportunity to introduce Professor P. Venugopalan. First of all, he is a very good and humble human being. He did his PhD from one of the premier institutes of India, ISC Bangalore, in 1991. And from 1991 to 1994, he went to Switzerland, University of Bonn, and did his first postdoctoral research. Then he moved to New York, USA, State University, and he did his second postdoc. Then he joined Punjab University in the year 1995, and after several years, he became the HOD of the Department of Chemistry, Punjab University, Chandigarh, and he served as chairman from 2014 to 2017. One more attribute he has, he is one of the best crystallographers in India. Actually, whoever is working on supramolecular chemistry, hydrogen bonding and all those interactions, he is one of the best crystallographer. And also he has published several papers. His articles are awesome. And he has more than 200 papers. He has collaboration from almost all IITs. So it is a great privilege for all of us to listen to him. He is from Kerala, so he is very much comfortable in English. But I am not forcing you, sir, but I request you for our BSc and MSc student, if a little bit Hindi you can say, it will be great. Otherwise, English is okay for us. Thank you, sir. Over to you, sir.
Sir, unmute yourself.
Your voice is not coming, sir. Your camera is on, you are visible, but your voice is not coming, sir. You are still not audible, sir.
Sir, unmute yourself.
You are mute, sir, from your side. Not audible, sir. Still not audible, sir. Okay, let me check if there is any setting change from this end. Still not audible, sir. We are extremely sorry because of the technical glitch. We cannot continue with the present lecture, so we will move on to the next speaker. Thank you.
Professor Nizamuddin, ex-head, Department of Chemistry, Deen Dayal Upadhyay, Gorakh University, Gorakhpur. I would like to invite Professor Nizamuddin sir to deliver the talk, please. Sir has done his MSc from Deen Dayal Upadhyay, Gorakh University, Gorakhpur in 1974. PhD from the same university in 1978. Postdoctorate fellow from East Carolina University, United States of America from 1982 to 84. He has guided 23 PhD research scholars. He has to his credit approximately 84 research papers published in international reputed journals with high impact factors. He has been permanent members of many societies. I would like to name one of them. Agricultural and Biological Society, Japan. He has been consultant of so many pharmaceutical industry, drug making company in India, abroad. Few of them is Pfizer, Wockhardt, Cipla, Ranbaxy, etc. I would call upon Professor R.K. Singh Sahab, the convener of the International Conference, to felicitate Professor Nizamuddin sir. Thank you.
Thank you, Professor Ansari, for statistical introduction. Good afternoon to all of you. Well, dear audience, I guess we might have enjoyed well so far with so many good lectures from different speakers. And the most of the speakers might have shared their research work before you. Since I am a retired person, so I don't have any current research data or result which I can produce before you. Therefore, I thought, why not take such a topic which is comparatively new and may be beneficial, at least for the students sitting here in this hall. Accordingly, I have decided to take the topic, the 2022 Chemistry Nobel Prize, that is click chemistry. Well, before going ahead, I must be very honest to say that I'm not master of this topic, because topic itself is Nobel Prize winner. So what I did, I read this subject several times, not 1, 2, 3 times, several times, and collected literatures from different places and have added my own teaching experiences to make this subject easier and easier so that it can be understood. That I am going to present before you. Now the first question, अभी रहने दीजिए उसी slide को please. Now the first question arises, what is click chemistry? At this stage, I can't define click chemistry, but I can only say that it is a chemical philosophy and given by K.B. Sharpless. You might have heard the name of K.B. Sharpless. Nobel laureate, he got Nobel Prize in 2001. So he is the initiator of this subject. What chemical philosophy he has given regarding this subject, that is based on certain cues that he has taken from nature. What cues he has taken from nature, you will see in the next slide. Next slide, please. Now I will cover these subjects regarding chemistry. Next slide. Okay. First slide is chemistry of life. You are a student of chemistry. You are studying chemistry of lanthanide, actinide, transmetal, carbonyl compound, etc. But you have not think about what is chemistry of life. So Jean Nicholas for the first time pointed out that certain elements, especially carbon, nitrogen and oxygen, when combined in nature, they give 3 class of compound. Next slide, please. दबाते रहिए. रुक जाइए. Carbon, hydrogen, oxygen, nitrogen, they combine in nature, they give 3 very important amino acids, nucleotides and sugars. They undergo biopolymerization in nature. They give 3 biopolymers, namely polypeptide, that is protein, polynucleotide and polysaccharide, that is sugar. दबाते रहिए, रुक जाइएगा. Now, these 3 class of compound are called as primary metabolite because they are responsible for the growth development of any living organism. So he called these 3 class of compound as molecule of life. So what is chemistry of life now? The presence of 3 class of molecules. Because suppose anybody don't have sugar in his body, what will happen? He will die after 1 month, 2 months, or after 1 year. Suppose he doesn't have sugar and also no protein, then what will happen? He will die more earlier. And if he doesn't have polynucleotide, also all 3, then he cannot survive. So life is due to the presence of 3 class of compound. That's why they are called as molecule of life. Now, these primary metabolites undergo further polymerization through ATP. They give secondary metabolites. They are very complex compounds like steroid, cholesterol, alkali, terpenite. They are secondary metabolites. They are very complex molecules. I have taken one such structure here. Next slide, please. What is nature's chemistry? Let us go back to the best friend nature. The same sequence, amino acid, nucleotide, and monosaccharide converted to this, and they converted to the secondary monomer. आप दबाते रहिए. Yes. How these are formed from monomers? These three polymers, how these three polymers are from monomers? By a reversible simple carbon heterobond formation. Look here. Carbon, carbon. All these have small units are linked together by carbon heterobond. Now, if you go back to this conversion of primary metabolite to secondary metabolite, they involve sophisticated carbon-carbon bond because they are very complex molecule and several carbon-carbon bonds are formed. So, what he observed this thing and included this thing. If you go through the structure of these three classes of compound, you will find they are formed by connecting small unit, 1 unit, 2 unit, 3 unit, 1 unit, 2 unit, 3 unit with carbon heterobond. First thing. Second thing, less than 4 carbon atom to be attached with heteroatom. कहीं से काम कीजिए चौथा या तीसरा कार्बन कार्बन हेटेरोबॉन्ड से जुड़ा हुआ है. नेक्स्ट कंटेन बैठ जाइए. अच्छा चलिए हाँ बैठिए. कंटेन्स मैनी कार्बन हेटेरोबॉन्ड देख लीजिए. रिवर्सिबल CX बॉन्ड फॉर्मेशन देखिए. पॉलीसेकेराइड मोनोसेकेराइड से पॉलीसेकेराइड बनता है. पॉलीसेकेराइड को हाइड्रोलाइज कीजिए तो क्या बन जाएगा? मोनोसेकेराइड. अमीनो एसिड से प्रोटीन बनती है, उसको हाइड्रोलाइज कर दिए फिर हम अल्फा अमीनो एसिड बन जाएंगे. तो रिवर्सिबल ये रिएक्शन है और रिएक्शन इज रिलायबल, फेल नहीं होती है कभी. वेरी इंपॉर्टेंट थिंग फॉर रिसर्च. सिंपल एंड मीडियम इज एक्वस. So thus nature is a combinatorial chemist. It makes diversity of bipolymer with carbon heterobond that you call as primary metabolite. Next slide please. Chemistry or drug discovery. What chemist said? He isolated some compound from natural products, then subjected to the biological screening. If it has some biological activity, then it changed the functional group and advertised. If some active compound is formed, then it was through the process chemistry. So from here to here, that takes at least 10, 12 years. Look here. I have taken one example, Tynamycin. Most potent natural antibiotic, nature से निकाली गई है. But it's developed some resistance against certain bacteria and microorganisms. So what alternative was this? People were looking for the alternative of this. They are able to synthesize this compound. This ethyl amino group had been changed by this. And this compound was found to be more active than Tynamycin. But you will be surprised to know this synthesis took 6 years. दबाते रहिए 6 साल में बना. Secondly, in the New Year, look here. This is acting against HIV first and second. If you see the structure, it has 5 chiral center. So 32 isomers are possible. Very difficult synthesis. You have to control the stereochemistry because only this isomer is active. 31 isomers are not active. So very tough synthesis. So what about that result is the drug discovery based on natural product synthesis requires time, money and difficult synthesis, and that to lead a very costly drug. Next slide please. So, what is in 3 slides से क्या मिला उनको? What cues he had taken? K-berry sulfur has taken this easily. Polysaccharide from monosaccharide. This is, and what he has observed that the conversion of these monomers to polymers is reliable stepwise pit with CX bond formation. Small units are joined reversibly. A medium is aqueous. So, keeping these in mind, Sasbes thought, why not develop a chemical method which describes chemistry to generate substances quickly and reliably by joining small units together as nature does. क्योंकि ये रिएक्शन रिलायबल है, जो सिंथेटिक केमिस्ट है उनको मालूम है कि अगर 10 स्टेप की सिंथेसिस है तो 9 स्टेप में आप फेल हो गए या 10 में तो बेकार गया सब, किसी काम का नहीं. तो रिएक्शन जो आप करते हैं रिलायबिलिटी होनी चाहिए कि हाँ होगा, इन भले कम आवे. नेक्स्ट स्लाइड प्लीज. तो केबी साल्वेंस पॉइंट हुआ कहा है. सो why do we spend time and money to synthesize secondary metabolite by difficult carbon-carbon bond formation? मना करते हैं कि difficult carbon-carbon bond formation मत कीजिए, अगर बनाना ही है तो intramolecularly. Very important point. Do what nature do easily and what is easier to do. Very important point for researcher. Next point. All researchers must be restricted to the molecules that are easy to make. Make easy molecule and look for potential. See in the Nobel laureate lecture what he said. The most fundamental and lasting objective of synthesis is आते रहते हैं एक सीरीज में 50 कंपाउंड 40 कंपाउंड नो प्रॉपर्टी. पेपर छाप लीजिए आप. डू यू नो कितने पेटेंट हैं इंडिया के इंडिपेंडेंस के बाद? सिर्फ 5 पेटेंट हैं, वो CTRI के हैं. इसमें से कोई नहीं शायद जानता होगा एक भी. 5 पेटेंट हैं CTRI के, ये एक बैठे हैं वो जानते हैं. उसके अलावा सेंटाक्लोमोन जो है एंटीबायोटिक एजेंट है, उसका भी नाम लोग नहीं जानते. तो पेपर आप छाप लीजिए, उससे कोई मतलब नहीं है. प्रॉपर्टी होनी चाहिए हमारे यूज की, हमारे आपकी. दैट इज इंपॉर्टेंट. नेक्स्ट स्लाइड प्लीज. थोड़ा टाइम लगेगा, आप कहिएगा तो हम कैप्चर कर लेंगे स्लाइड को. हाँ, तो कॉन्सेप्ट ऑफ प्लिस केमिस्ट्री आफ्टर केमिकल फिलॉसफी बाय केबी साल्वर्स इन 2001 2001 में, और नोबेल प्राइज मिली है 2022 में, 21 साल काम करने के बाद. जैसे में it describes chemistry tailored to generate substances and quickly and reliably by joining small units together. छोटी छोटी यूनिट को आपस में जोड़ने के लिए जो आसानी से जुड़ सके और रिलायबल हो. नेक्स्ट, दिस इज इंस्पायर्ड. ये कहां से इंस्पायर हुआ? दिस इज इंस्पायर्ड बाय द फैक्ट that nature also generates substances by joining small modular units. Next, click chemistry, not a specific reaction. It is a concept that mimics nature. As defined by K.V. Sharpless, click chemistry, a set of powerful, virtually 100% reliable, selective reactions for the rapid synthesis of new compound via carbon heterolinkage. This is the definition. Click chemistry is a set of powerful, selective and modular blocks that work reliably in both small and large. जिसे पेटेंट करने में बहुत सारे रिएक्शन लैब में हो जाते हैं, और अगर आप उनको मास स्केल पर करें तो फेल कर जाते हैं. तो ये भी एक क्राइटेरिया इसके लिए होनी चाहिए. नेक्स्ट स्लाइड प्लीज. These are the guys who got Nobel Prize for the fundamental development of click and bioorthogonal chemistry. This lady, young lady, look at here, young lady, born in 1966. कितनी इसकी उम्र हो गई इस वक्त? लगभग 60 साल, है ना? 60 से कम ही है. This, हाँ, कुछ कीजिए. Mendel, Martin Mendel, 70 years. And this guy, K.B. Sharpless. This is the only guy who got twice Nobel Prize in chemistry. Once 2001, once 2022. Can you identify? This is the lady Where is KB Sarbless in between? इसमें कौन KB Sarbless है? He doesn't have hair on his head, so you can judge this one. This is KB Sarbless with mortal. Yeah, I have put this slide only because this is the guy who initiated the reaction, but he failed to explore this reaction. So he was unlucky. He has not got Nobel Prize, but he is the initiator, real initiator. Next. थोड़ी चिकने भी सार्वजनिक के लिए देखिए 2001 में नोबेल प्राइज इसको मिली है किस पे? ये 4 रिएक्शन लिखे हैं। 1970 में एसिमेट्रिक शार्पलेस एसिमेट्रिक डाइहाइड्रोक्सिलेशन, शार्पलेस एसिमेट्रिक इपॉक्सिडेशन, शार्पलेस एसिमेट्रिक मोनोहाइड्रेट, ये एमएससी के लड़कों को पढ़ाया जाता है, पढ़ते होंगे। इस पे नोबेल प्राइज मिली थी 2001 में। नेक्स्ट प्लीज। तो क्राइटेरिया, किस क्या क्राइटेरिया होनी चाहिए किसी केमिकल की रिएक्शन के लिए? वो क्लिक रिएक्शन कहलाए। जैसे आप कहते हैं कि कोई सिस्टम आयन या न्यूट्रल मॉलिक्यूल हेटेरोसाइड एरोमेटिक कब होगा? when he fulfills some criteria. So a chemical reaction said to be the click reaction if it fulfills the following criteria: be 100% reliable, quick, simple and wide in scope. बड़ी खास प्रॉपर्टीज हैं। Be very high atom economic and high yield. इससे क्यों होता है? क्योंकि जो ड्रग मार्केट में आती है अगर उसकी यील्ड कमजोर है तो कास्ट उसी हिसाब से बढ़ जाएगी। स्टार्टिंग मटेरियल अगर बहुत महंगे हैं, स्टेप बहुत लंबी है तो कास्ट उसकी बढ़ती चली जाएगी। बी स्टीरियो स्पेसिफिक एंड बी फिजिकली स्टेबल। नेक्स्ट स्लाइड प्लीज। आगे क्लास ऑफ क्लिक रिएक्शन। मैंने एमएससी के बीएससी के स्टूडेंट के लिए सिर्फ ऐसे रिएक्शन चूज किए हैं जिसको वो जानते हैं। तो पहला है क्लिक रिएक्शन, एडिशन टू कार्बन कार्बन मल्टीपल बॉन्ड। क्या होता है इसमें? सब का सब प्रोडक्ट में जुड़ जाता है। एडिशन रिएक्शन कौन सी रिएक्शन है? जिसमें रिएक्टेंट के सारे एटम प्रोडक्ट में इनकॉर्पोरेट हो जाए तो ये 100% हो गई। तो न्यूक्लियोफिलिक सब्स्टिट्यूशन, सारे न्यूक्लियोफिलिक्स नहीं, रिंग ओपनिंग ऑफ इपॉक्साइड 3 मेंबर रिंग। 3 मेंबर रिंग क्या है? हाईली स्ट्रेनलेस रिंग है, इसलिए बड़ी जल्दी टूटती है। और क्या है? 1,3 डाइपोलर साइक्लो, इसको हम लेंगे अभी। एंड रिवर्सिबल कार्बोनिल केमिस्ट्री, एस्टर फॉर्मेशन, केचेट एक्सेट्रा, और कार्बोनिल केमिस्ट्री जिसमें नॉन अल्डोल हो, ऑक्सोन हाइड्रोजन फॉर्मेशन एक्सेट्रा। दे आर द सिंपल रिएक्शन दैट हैव बिकम दे आर क्लिक रिएक्शन। नेक्स्ट प्लीज। I have selected few reactions. They are very important click reaction, and people have already worked and working on these reactions. So first reaction is this alkyne and azide. Treat with copper catalyst that give 1, 2, 3, 1, 4 disubstitute 1, 3, 1, 2, 3 triazole. That is called as CUAC reaction. Copper catalyzed alkyne azide cycloaddition reaction. Next please. The same reaction, but no catalyst. Alkyne is in the ring. Here open chain, no catalyst. Here catalyst is needed when alkyne is open chain, but catalyst is not needed when it is in ring. I will take this letter. This is called as strain promoted alkyne azide cycloaddition reaction. Next please. This is D-Lender reaction, typical D-Lender that you know already. Next. दिस इज थाइन एलिन रिएक्शन। ये 4 रिएक्शन है जिसमें बहुत काम वर्क हो रहा है और इन दोनों पे पहले, दूसरे और तीसरी पर पीपल आर वर्किंग ऑन दिस एरिया। नेक्स्ट प्लीज। थोड़ा सा हम एक स्लाइड इसके लेंगे। रिसेल्डर रिएक्शन जानते हैं, एक डाइन और डाइनोफाइल जब आपस में रिएक्ट करती हैं तो 4 + 2 बाई साइक्लोएडिशन होता है। दुनिया की ये बहुत पुरानी रिएक्शन है, 1954 में है, लेकिन आज तक कोई रिएक्शन नहीं पैदा हुई, अभी तक किसी ने नहीं निकाला कि जिसमें एक सिंगल स्टेप में 4 कायरल कार्बन आप रिएक्ट करते होंगे और डिफरेंट स्टीरियोकेमिस्ट्री हो। सिर्फ एक रिएक्शन है जो थोड़ा टच कर रहा है, रॉबिन्सन एनुलेशन, जिसमें 3 कायरल कार्बन आप क्रिएट कर सकते हैं। सो दिस इज द ब्यूटी ऑफ द रिएक्शन, दैट व्हाई इट इज स्टिल वर्क, पीपल आर वर्किंग। नेक्स्ट प्लीज। थोड़ा सा हमने इसमें ध्यान दबाते रहिए। मेरे पास टाइम नहीं है ना, मैं डी-सेल्डर रिएक्शन पर हाथ दूंगा तो आप समझते हैं हाँ कि क्या है, कौन सी बला है। आप चाहते हैं डिसल्टर रिएक्शन बोलने चले आए। देखिए कैसे स्टीरियोकेमिस्ट्री हमने दिखाया है कैसे हो। देखिए बिल्कुल आप देखें कौन कौन सी से, देखिए शायद एमएससी के स्टूडेंट है, भले हम पूरी ना बोल पाए लेकिन उनके फायदे के लिए। लुक हियर, जब डाइन अप्रोच करेगी डाइनोफाइल डाइन के बाद तो ये सिचुएशन बनेगी। अब गौर से देखिए डाइन का जी और इसका भी, ये दोनों पास पास है, एक साइड में है, ये दोनों एक साइड में, तो ये दोनों शीश हो जाएंगे, ये दोनों शीश हो जाएंगे, ये दोनों शीश, प्रोडक्ट निकलना आसान हो जाएगा। नेक्स्ट प्लीज। जनरेशन कैसे आप CX बांड जनरेट करेंगे, तो हमने कहा ना न्यूक्लियोफिलिक रिंग ओपनिंग ऑफ स्प्रिंग लोडेड रिंग्स, 3 मेंबर रिंग बहुत हाईली स्ट्रेनस है, इनको तोड़ने में बड़ी आसानी है, तो आप इसको तोड़ सकते हैं। आप एक बार पुश कीजिए, देखिए इसी में आपके एपॉक्साइड भी हो जाएगा, रेनिन भी हो जाएगा, सब इसमें बन जाएंगे। और इसको न्यूक्लियोफाइल से तोड़ेंगे तो क्या होगा, ये बन जाएगा। लुक हियर, 2 कार्बन हेटेरो बॉन्ड, कार्बन ऑक्सीजन एंड कार्बन इन सिंगल स्टेबिक कार्बन, and these reactions are reliable, stereo specific, mainly regio selective and quantitative, so they are reaction। हाँ आगे बढ़ाते जाइए, इसको हम नहीं लेंगे, तो समझ गए लोग, वही है ऑक्साइड बनाना। हाँ रुके, I will take only this reaction, 1,3-dipolar cycloaddition of azide and alkyne or with hydrogen। वो गाई जो बर्टोली के साथ बैठे हुए हैं, हाथ कंधे पर हाथ रखे हुए, he discovered this reaction। What he taken? 1,3-dipole of the type ABC, this should be linear, not linear, non-linear, that reacts with dipolarophile like alkene or alkyne under 3+2 cycloaddition give 5 number heterocyclic, especially 1, 2, 3 triazole। Here is 1,3-dipole, look here, नेगेटिव चार्ज पॉजिटिव चार्ज, इसको शिफ्ट कर दिया यहाँ तो ये बन जाएगा। तो बी हमेशा पॉजिटिव है, ए सी क्या है, नेगेटिव है, तो डाइपोल है। और ये अल्काइन है, अल्काइन को या अल्कीन को आप ब्रेक करेंगे तो एक कार्बन पर पॉजिटिव, एक पॉजिटिव चार्ज होगा, एक पर नेगेटिव, फिर दूसरे पर वही उलट देंगे, ये डाइपोलरोफाइल हुआ, एक ही कार्बन पर पॉजिटिव भी और नेगेटिव भी चार्ज हो, ये दोनों आपस में रिएक्ट करेंगे तो 5 मेंबर रिंग कंपाउंड कैन बी। आप प्रेस कीजिए प्लीज। 2 एग्जाम्पल बहुत अच्छे हैं, डाइजोमीथेन है और ये है एजाइड आयन, ये आर वेरी गुड एग्जाम्पल, और भी हैं आप इसकी। एजाइड आयन एंड एल्काइन दोनों रिएक्ट करेंगे आपस में, 3+2 5, ये ट्राइazole, 1, 2, 3 ट्राइazolene बन गई। हाँ पुश कीजिए एक बार। दिस इज Hussein reaction, thermal Hussein reaction। Alkyne reacts with the azide ion at 80 degree thermally, that gives a mixture of 1,4-disubstituted 1, 2, 3 triazole and 1,5-disubstituted 1, 2, 3 triazole, 2 products। Of course this is more ill than this, this is lesser, this is major। Next please, mechanism of Hussein reaction। Just I am introducing this subject to you, just you imagine, right, only this। So this is a pericyclic reaction। Pericyclic reaction in which molecular orbital of 1,3-dipole giving 4 pi electron reacts with dipolar giving 2 pi electron, undergo cycloaddition reaction, give heterocyclic system। That is the total mechanism of Hussein reaction। I have taken this general reaction, you have seen that when alkyne azide react at 30 degree Hussein reaction, 2 products are isolated, 1, 4 substituted, 1, 5 substituted। कुछ प्लीज मैकेनिज्म हमने कोशिश किया, देखिए ये दोनों को क्या किया है हमने, एजाइड आयन ये है, जैसे लिखा है हमने लिख दिया, और ये एल्काइन है, इसको हमने एक बांड को तोड़ दिया, पॉजिटिव और नेगेटिव। एल्काइन क्या है, न्यूक्लियोफाइल, और एजाइड आयन क्या है, इलेक्ट्रोफाइल। देखिए नाइट्रोजन पॉजिटिव चार्ज है, तो क्या है, दिस नेगेटिव चार्ज शिफ्ट हुआ, दिस शिफ्ट हुआ एंड दिस शिफ्ट हुआ, ये मिल जाएगा। दूसरे में उलट दीजिए इसको उलट के जैसे में कर दीजिए तो ये 1,4 बन जाएगा। और इन बोथ ऑफ दिस 3 एटम एक के, 2 एटम एक के, 5 एटम आपस में रिएक्ट करते हैं 5 नंबर रिंग। नेक्स्ट स्लाइड प्लीज। फ्रंटियर मॉलिक्यूलर ऑर्बिटल ट्रीटमेंट ऑफ दिस रिएक्शन। एजाइड में 3 sp2 नाइट्रोजन है, राइट, यू हैव 3 मॉलिक्यूलर ऑर्बिटल। 3 मॉलिक्यूलर ऑर्बिटल में क्या होगा, एक बाइंडिंग होगा, एक उसका एंटी बाइंडिंग होगा, और एक क्या होगा, नॉन बाइंडिंग होगा। देखिए गौर से देखिए, ये नॉन बाइंडिंग इलेक्ट्रॉन है, ये पाई बॉन्ड है। तो दो इलेक्ट्रॉन यहाँ, दो इलेक्ट्रॉन, ये बाइंडिंग है। सारे एक तरह के लोब एक साइड में और दूसरे तरह के दूसरे साइड में। नो नोडल पॉइंट इन दिस इज एंटी बाइंडिंग ऑफ दिस एंड दिस इज नॉन बाइंडिंग। इस तरह से बाइंडिंग है। तो दो इलेक्ट्रॉन इसमें, दो इलेक्ट्रॉन, 4 इलेक्ट्रॉन हो गए। ये हो गया आपका 1,3 एक बार और पूछिए। दिस इज मॉलिक्यूलर ऑर्बिटल डाइफ्लोरोफाइल। तो इथाइलिन में दो ही मॉलिक्यूलर ऑर्बिटल है, एक बाइंडिंग और एक एंटी बाइंडिंग। तो फ्रंटियर मॉलिक्यूलर ऑर्बिटल आप जानते होंगे किसको कहते हैं, जिसमें जो रिएक्टिंग सिस्टम है, the molecular orbital of reacting system are converting to each other. But this the conversion is not random. Only those molecular orbital of reactant are converting into molecular of the product, in which there is a certain symmetry common between these two. If this occurs, then the transition state form is of lower energy and reaction is said to be symmetry allowed. Otherwise, it is forbidden. Look here. अब फ्रंटियर ऑर्बिटल देखिए, ऑक्युपाइड मॉलिक्यूलर ऑर्बिटल है, ये अनऑक्युपाइड मॉलिक्यूलर, और एक ये ऑक्युपाइड मॉलिक्यूलर, और ये भी ऑक्युपाइड है, दो ऑक्युपाइड मॉलिक्यूलर है। इसकी एनर्जी ज्यादा है तो इसको हम कहेंगे हाईएस्ट ऑक्युपाइड मॉलिक्यूलर ऑर्बिटल। इसकी एनर्जी कम है तो वो लोएस्ट ऑक्युपाइड मॉलिक्यूलर, और ये अनऑक्युपाइड है, एक ही है, तो यही ल्यूमो भी हो जाएगा, ये होमो। तो जो दोनों रिएक्टिव पार्टनर के होमो और ल्यूमो होते हैं, दे आर कॉल्ड एज फ्रंटियर मॉलिक्यूलर ऑर्बिटल and reaction occur in between these 4 orbitals. तो look here. हाँ, प्रेस कीजिए एक बार प्लीज, प्रेस। हाँ देखिए, ठीक। अब देखिए, एक का होमो दूसरे का लोमो, दूसरे का होमो और पहले का लोमो, होमो लंबो 2 इंटरेक्शन पॉसिबल है। अब ये जो ब्लू कलर से है, इसकी एनर्जी डिफरेंस मिनिमम है, so this will be more favorable than the red ones. प्रेस कीजिए प्लीज, और प्रेस कीजिए। मोड ऑफ एक्शन, तो होमो देखिए, ये क्या है, होमो है एजाइड आयन का, और ये क्या है, ल्यूमो है एल्काइन का। दे विल देखिए सिमिलर लोब कैन overlap to each other in the same phase, so overlapping is suprafacial. And since it gives 4 pi electron, it gives 2 pi electron, so that we can write 4 pi to the power 4s and pi square 4s. इसका उल्टा कर दीजिए तो भी ये रिजल्ट मिलता है, तो ये सुपरफिशियल है। नेक्स्ट, मॉडिफाइड ह्यूसन रिएक्शन। ह्यूसन रिएक्शन को अब यहाँ से लोगों ने काम करना शुरू किया, ह्यूसन की चीज को आगे बढ़ाना शुरू किया। उन्होंने फॉर्मली किया था, इन्होंने कहा, ये मार्टिन मॉन्डल जो बीच में शख्स था कनाडा वाला, उसने पहली बार एक कॉन्फ्रेंस में इसने प्रपोज किया कि ह्यूसन रिएक्शन कैन बी कैरीड आउट इन प्रेजेंस ऑफ कॉपर 1 कैटलिस्ट। सो ही कैरीड दिस रिएक्शन इन कॉपर सल्फेट सोडियम एस्कार्बोरेट इन वाटर और टर्शियरी ब्यूटेनॉल 1,1 मिक्सचर। So he got this 1,4-triazole only. Instead of getting 2 products, 1,4 and 1,5, he got only 1,4-triazole. Right? What is function of? देखिए कॉपर इसमें सेकंड स्टेट में है, कॉपर सल्फेट में। सोडियम एस्कार्बोरेट रिड्यूसिंग एजेंट है, कॉपर सल्फेट को कॉपर 2 को कॉपर 1 में कन्वर्ट करता है। दैट्स ऑल। हाँ, तो प्रेस कीजिए, ये जनरल रिएक्शन है। Then they generalize this reaction. Any azide or any terminal alkyne can react in this in presence of this catalyst. They give only this exclusively. The first reaction which was carried out in this regard was this, and now they call this reaction as Hussen's copper catalyzed alkyne azide cycloaddition, abbreviated by CUAAC reaction. Now this reaction is known by only this term, CUAAC reaction. Next please. Regioselectivity. देखिए, ये पहला है, हुसैन ने किया था, 2 प्रोडक्ट मिले, हाँ दबाते जाइए, फिर इसको कॉपर 1 कैटलिस्ट से एक ही 1,4 मिला, फिर रुदेनियम कैटलिस्ट से 1,5,3 मिलता है एक्सप्लोसिवली। तो दोनों का मिक्सचर बना पहले बार 1,4 1,5 दोनों आपने कर दिया. नेक्स्ट, now recently they have prepared Grignard reagent of alkyne. They have reacted with azide ion and they found this nucleophile Grignard reagent. And if you treat this one electrophile Rx, you will get tri-substituted. They are di-substituted and this is tri-substituted 1, 2, 3 triazole. Next please. Improved CUA reaction. ये तो था कि कॉपर से, अब ये हो गया कि कॉपर के अलावा तमाम सारे मेटल अब यूज कर लिए गए और उससे भी रिएक्शन और बढ़िया होने लगी है. तो क्या कौन कौन से हैं मेटल? ये है निकेल का, गोल्ड, सिल्वर, इरेडियम, लैंथनम, जिंक, कॉपर और रुथेनियम. They, if you treat this azide ion with this alkyne, so this reaction is called as metal catalyzed alkyne azide cycloaddition. You can get this triazole without any catalyst. Next, क्या करेक्टरिस्टिक है इसकी? अभी हमने 4-5 को बताया था. ये एक ऐसी रिएक्शन है जो सारे करेक्टरिस्टिक्स फॉर्म करती है. सारे करेक्टरिस्टिक्स जिसमें होने चाहिए क्लिक रिएक्शन के लिए वो सब इसमें है. So people call this reaction as champion click reaction or cream of the crop. Next, catalyst free azide alkyne cycloaddition. कैटलिस्ट इसमें ना हुआ. इसकी नीड क्या हुई? नीड ये हुई कि CUA रिएक्शन फोकस द इंटरेस्ट ऑफ इट्स यूज इन बायोलॉजिकल सिस्टम. Then the toxicity of copper ion to cell and living organism limits the use of this reaction in biological system. So that was a challenge before the scientist. Ultimately, the lady Bertozzi, he solved this problem. What she did? She took the alkyne not open chain but in the ring, an 8-member ring. So what will happen? अगर आप अल्काइन की ज्योमेट्री क्या है? लीनियर है ना. बार एंगल कितना है? 180 डिग्री. तो रिंग में 180 डिग्री बार एंगल हो ही नहीं सकता, तो हाईली स्ट्रेनस हो गई. तो इससे क्या होगा कि ये जल्दी से रिएक्शन करके अपने स्ट्रेन को खत्म करेगी. और सी फाउंड गुड रिएक्शन बिना कैटलिस्ट के हुआ और 10 to the power 7 times fast reaction हुआ. So this anxiety placing the alkyne into ring is a solution to CU reaction in biological system. This is azide. This अब ये जो शेकर जो हमने लिख दिया है ऐसा पॉसिबल नहीं है. क्यों? क्योंकि ये ज्योमेट्री 180 डिग्री नहीं है. एक बार और पूछ कीजिए, मे बी लाइक दिस. अगर इस तरह से है तो कितने बांध एंगल? 180 डिग्री, ये 90, 90, 3 तरह के बांध एंगल इसमें आ गए. हाईली स्ट्रेन. सो दिस वर्क्स बेटर. कुछ कर दिए. You can catalyst this reaction. So they call this strain promoted azide alkyne cycloaddition, SPAAC reaction. एक बार और क्या है? क्या है इसमें? कैटलिस्ट फ्री है, नॉन टॉक्सिक मेटल है, इनहांस रेड है. चलिए नेक्स्ट, मैकेनिज्म. इसके पहले हम लोगों ने देखा था तो वो क्या थी? पेरिसाइक्लिक, थर्मल थी, और ये रूम टेम्परेचर पर हो रही है. दिस इज नॉट पेरिसाइक्लिक, स्टेपवाइज मैकेनिज्म है. आपके पास क्या है? यू हैव अल्काइन, एजाइड एंड कॉपर सल्फेट, सोडियम एस्कॉर्बेट. दैट्स ऑल. सो टर्मिनल अल्काइन, एसिडिक हाइड्रोजन that will react with copper sulfate, that will give copper acetylide. Simple reaction. Second, this copper acetylide react with azide ion in this fraction, and 3+2 you have this triazole cuprate. Deep now protection of this copper that gives the triazole. 1,4 disubstituted triazole. Next, please. Catalytic cycling. You have this reaction: azide, alkyne, copper sulfate, sodium phosphate. This catalytic cycling क्या है? ये दोनों आपस में रिएक्ट करेंगे. सोडियम कॉपर एसिटाइड बन गया. Once more, please. Then copper again react with this alkyne, dicuprate. Next, please. Next, आपने दो चीज कर लिया, कॉपर और अल्काइन को हिट करके ये बना लिया. एजाइड आयन आपके पास है. This negative charge will attack over here, and you will get this, and this again converts into cyclic ring, 6-member ring. This undergo rearrangement by breaking this bond over here and copper emulates, and you are getting this. And ultimately, a proton is taken by this copper from the alkyne and right product, and again this generated, and this cycle is continued. Next prize, bioorthogonal chemistry. दोनों पर मिला है प्राइज, नोबेल प्राइज, केमिस्ट्री एंड बायोऑर्थोगोनल केमिस्ट्री क्या है? A set of reaction that can take place in biological environment without affecting biomolecule or interfering with the biochemical process. बायोलॉजिकल इन्वायरमेंट में क्लिक रिएक्शन को अगर आप कर ले जाए तो यह है बायोऑर्थोगोनल केमिस्ट्री। कहां से लिया इसको? ऑर्थोगोनलिटी मैथमेटिक्स से लिया। लुक हियर, एजाइल लेवल्ड बायोमॉलिक्यूल है और साइक्लोऑक्टाइन लेवल्ड प्रोब है। क्लिक केमिस्ट्री करेंगे ये मिल जाएगा। नेक्स्ट, these techniques have been used in a number of sectors including diversity treatment that can kill cancer cell without perturbing healthy cells. कैंसर का पेशेंट धीरे धीरे वीक होकर कम मर जाता है क्योंकि जो दवा है वो कैंसर सेल के साथ नॉर्मल हेल्दी सेल को भी किल करती है। तो दिस इज बिग प्रॉब्लम। नेक्स्ट, फर्स्ट क्लिक केमिस्ट्री बेस्ड ड्रग, ये मार्केट में आने वाला है, थर्ड स्टेज क्लिनिकल ट्रायल में है और इसी बर्स्टोजी के देखने में एक केमिकल बन रहा है, एंटी कैंसर एजेंट है, अभी डिस्क्लोज नहीं किया है। नेक्स्ट प्लीज, एप्लीकेशंस, तमाम सारे एप्लीकेशन है, मैं एक लूंगा यही 1 2 3 ट्राइजोल सिंथेसिस। नेक्स्ट प्लीज, आप जब टाइम हो जाएगा तो बता दीजिएगा। उसे 42 स्लाइड हमने किया है, बाकी काट दिया है। हाँ तो काटते हैं, चलिए बढ़ते रहिए। ये ट्राइजोल की बहुत सारे, दबाते रहिए, बहुत सारी एक्टिविटी ट्राइजोल की है जो हमने मेंशन किया है, कितना एक्टिव है। ह्यूमन लाइफ इज प्रोबेबली अनटच्ड विद द एप्लीकेशन ऑफ दिस। कोई जिंदगी, हमारी जिंदगी का कोई सुबह ऐसा नहीं है जहां ट्राइजोल की केमिस्ट्री जो है इस्तेमाल ना होता हो, हर फील्ड में है। नेक्स्ट प्लीज, दिस इज इंपॉर्टेंट। 1 2 3 ट्राइजोल हो गया ये एज बायोएस्टर्स ऑफ पेप्टाइड, मतलब दोनों की बायोलॉजिकल एक्टिविटी करीब करीब एक जैसी है। तो जहां कहीं एमाइड बांड है वहां 1 2 3 ट्राइजोल जोड़ दें तो देखें उसकी एक्टिविटी में क्या फर्क पड़ता है। देखिए हमने कोशिश किया करने के लिए, देखिए ये नाल बाइंडिंग इलेक्ट्रॉन, ये नाम है हाइड्रोजन बॉन्ड डोनर, हाथ दबाइए, ये दीजिए। ये ऐसा कार्बन है जो इलेक्ट्रोफिलिक है, कार्बन ऑक्सीजन बोलवा, कार्बन नाइट्रोजन के साथ जुड़ा है। नेक्स्ट, ये देखिए हाइड्रोजन है, हाइड्रोजन बॉन्ड डोनर है, हाइड्रोजन बॉन्डिंग में ले। नेक्स्ट, ये देखिए डिस्टेंस R1R की डिस्टेंस जो है 3.4 एंगस्ट्रॉम है, इसमें 3.9 सिमिलर है। नेक्स्ट, ये देखिए डायपोल मोमेंट, इसका 4.1 डिवाई है, इसका 4.6 डिवाई है। तो बहुत सारी फिजिकल प्रॉपर्टी दोनों की मिलती जुलती है, इसलिए ओवरऑल ट्राइजोल कैन बी पोटेंशियल सरोगेट ऑफ हेमाइड बॉन्ड्स फॉर पेप्टाइड मॉडिफिकेशन। नेक्स्ट प्लीज, अब देखिए क्या यूज है इसका। यहां हमने एक एग्जांपल लिया है, ये एक ड्रग है एम्प्रेमिनेविर, बहुत इंपॉर्टेंट है। ये 19 पीछे जाइए 1997 से ये क्लिनिकल यूज में है। इसमें देखिए एक एमाइड बांड है, तो उन लोगों ने क्या किया कि इस एमाइड बांड को 1 2 3 ट्राइजोल से रिप्लेस किया और इस पूरे पोर्शन को इससे रिप्लेस किया। एक कंपाउंड ये मिल गया, दूसरे में इसको इससे रिप्लेस किया। ये तो नहीं, लेकिन this compound was found to be more potent than first, so this is also is coming out in the market. Next, synthesis of tyrosine inhibitor। हाँ, ये देखिए ये टेट्रा पेप्टाइड है, 4 अमाइनो एसिड से साइक्लिक है, साइक्लिक टेट्रा पेप्टाइड। प्रोलीन है 2 मॉलिक्यूल, एक टायरोसिन है ब्लू कलर से और एक वायलिन है। अब देखिए इसमें एक एमाइड लिंकेज यहाँ ओपन है और एक यहाँ दो ओपन है। इन दोनों एमाइड लिंकेज को दे है चेंज विद ट्राइजोल। पुश कीजिए प्लीज, ये बहुत एक्टिव है, पहले से यूज हुआ है। देखिए एक को चेंज किया, इसको इसमें इसको चेंज किया और इसमें दोनों को चेंज किया, और जब दोनों को चेंज किया तो इट इज फार बेटर देन दिस बिकॉज़ IC50 is 1.5 and this is 1.1। नेक्स्ट, सिंथेसिस ऑफ एंटीफंगल फिनोल, बहुत इंपॉर्टेंट एंटीफंगल एजेंट है, आप टीच किए होंगे। ये FDA अप्रूव्ड एंटीफंगल ड्रग है। इसमें जो है ट्राइजोल वो 1 2 3 नहीं है, 1 2 4 ट्राइजोल के 2 आइसोमर है। देखिए ये है 1 2 3 ट्राइजोल और ये है 1 1,2,4 ट्राइजोल। तो इसमें 1,2,4 ट्राइजोल है। लोगों ने सोचा कि इन दोनों को चेंज कर दें 1,3 से 1,2,3 से, या एक को चेंज कर दें 1,3। तो जो एक्टिव हुआ वो मैंने लिया है। इसमें देखिए इन्होंने क्या लिया, एजाइड ले लिया, जिसकी जगह पे एजाइड अल्काइन ले लिया, एजाइड दे ट्रीटेड विद एजाइड एंड दे गॉट दिस कंपाउंड। इन सेकंड केस they treated this azide here in place of alkyne azide, and here is alkyne, and they got this compound, and both of these were found to be active on vaginal candidiasis। बहुत जबरदस्त फंगस के लिए दोनों यूज किए जाते हैं। नेक्स्ट एग्जाम्पल ऑफ क्लिक सीक्वेंस डाइवर्सिटी हुई। एक एग्जाम्पल मैं लूंगा कि 4 स्टेप एक सिंगल पार्ट में आप करेंगे और कितने बढ़िया से हुआ है। हमने रुक जाए, रुक हियर, ये क्या है, 1,4 साइक्लोहेक्सेन डाइहेक्साडाइन, 1,4 साइक्लोहेक्साडाइन। इफ आप ऑक्सीडेशन करेंगे तो हमने दोनों सिस लिया है, तो ये सिस बन सकता है, दोनों ट्रांस हो सकता है। एक ही जा हुआ, एक सिस हो सकता है, एक ट्रांस होता है, बाद में हम लेंगे। इससे 4 ग्राम लिया तो 5 ग्राम ये मिल गया। इसको एजाइड आयन प्री मेमोरिंग है, स्प्रिंग लोडेड है, एजाइड आयन बड़ी आसानी से खोल देगा। इसको इन्होंने एजाइड आयन से खोला तो ये बन गया। अब देखिए इस कंपाउंड से इसमें 4 कार्बन हेटेरो बॉन्ड है, 2 कार्बन ऑक्सीजन 2 कार्बन नाइट्रोजन। एजाइड मिल गया तो अल्काइन से रिएक्ट करके ट्राइजोल दो बना दिया। अब ये फिर इस पे डिहाइड्रेशन कर दिया तो ये बन गया ये कंपाउंड। आप 4 ग्राम से चले थे और 17.5 ग्राम आपको मिल गया, और सब एक पार्ट में किया गया है ये रिएक्शन। नेक्स्ट इसका दूसरा पार्ट ले लिए, ये दोनों सिस थी, दे आर ट्रांस एंड सिमिलर रिएक्शन वे कैरीड आउट। क्या होगा, स्टीरियोकेमिस्ट्री दोनों की बदल जाएगी। इसमें दोनों सिस होंगे और इसमें दोनों ट्रांस हो जाएंगे ट्राइजोल। तो दिस इज कॉम्प्लेक्स स्ट्रक्चर कैन बी इजीली रैपिडली असेंबल्ड बाय क्विक केमिस्ट्री। ट्राइसाइक्लिक कंपाउंड एक्स एंड वाई देखिए ये इस तरह के हैं, स्ट्राई टाइप के, आर प्रिपेयर्ड इन वन पास 3 सीक्वेंशियल क्लिक रिएक्शन। नेक्स्ट प्लीज, यील्ड है ये। नेक्स्ट प्लीज, चलिए 2 बार आ रहा है फिर से। डिसल्टर क्लिक इन ड्राइमर क्या है, ट्री लाइक कंपाउंड, बड़ा यूज है इनका, हमने यूज यहां दिया हुआ। एक हम एग्जाम्पल लेंगे, ये देखिए अल्काइन है और ये क्या है 1,3 साइक्लोपेंटाडाइनोन। ये डाइन है, ये डाइनोफाइल है। डिसल्टर रिएक्शन होगा तो ये देखिए 6 मेंबर रिंग बन गई, और रिट्रो डिसल्टर होगा, कार्बन डाइऑक्साइड निकल जाएगी तो ये मिल गया। 4 बेंजीन से चले थे, 1 बेंजीन बन गई, 5 बेंजीन, 6 बेंजीन मिल गई ये डिसल्टर से। अब इसको उन्होंने मोनोमर मान लिया, इसके बाद डेंड्राइमर बनाया। नेक्स्ट प्लीज, अब खत्म हो जाएगा, डोंट वरी। अब देखिए ये आपने पीछे बनाया है, यहाँ बाइफिनाइल सिस्टम लिया है, 4 अल्काइन लिया, 4 मॉलिक्यूल ले लेंगे तो क्या होगा, ये 4 रिंग बन जाएंगे। इसमें देखिए इस तरह के आपने ले लिया, 4 ले लेंगे तो बनेंगे। इसमें 6 ले लिया, इसको 6 मॉलिक्यूल लेंगे तो ये डेंड्राइमर कैन बी। नेक्स्ट प्लीज, सिंथेसिस ऑफ एबीसी टाइप लीनियर, छोड़ दीजिए इसको क्योंकि लोग भूखे हैं। हाँ मैं समझ रहा हूं। नेक्स्ट प्लीज, अब टाइम हम देख नहीं रहे लेकिन अब ज्यादा नहीं रह गए, 4 स्लाइड होंगे। सुगर ट्राइजोल कॉन्जुगेट वाई माइक्रोसाइज, लुक हियर, दिस इज एजाइड सुगर। इसमें एजाइड आयन भी है, अल्काइन आयन है। आप इसके 3 मॉलिक्यूल ले लीजिए और आमने सामने एक का एजाइड दूसरा का अल्काइन लिख लीजिए तो यू कैन गेट दिस सुगर कंटेनिंग ट्राइजोल। अगर n=2 ले लीजिए तो ये बन जाएगा। नेक्स्ट n=3 के लिए तो पीपल हैव प्रिपेयर्ड दिस माइक्रोसाइक्लिनाइजेशन इंक्लूडिंग एजाइड एंड सुगर यूजिंग क्लिक केमिस्ट्री। नेक्स्ट, सिंथेसिस ऑफ ट्राइजोल लिंक डीऑक्सीनाजोरिमाइसिन कॉन्जुगेट, बड़ा इंपॉर्टेंट ड्रग है ये। देखिए ये है अल्काइन और ये है सुगर एजाइड, अमाइनो सुगर है, डीऑक्सीनाजोरिमाइसिन एक्टिव कंपाउंड है। इसको बनाना है तो क्या करेंगे, इससे प्रोपार ग्लूटामाइड से डाइएस्टर में कर लेंगे, ये बन जाएगा। एक बार और पूछ कीजिए, क्विक रिएक्शन करेंगे, 4 मॉलिक्यूल ये से लग जाएगा। ये दिस सिंथेसाइज्ड कंपाउंड इज यूज्ड इन गोचर डिजीज, गोचर डिजीज जो हेरिडिटी से आते हैं माँ बाप के बच्चों में। नेक्स्ट, कैलेक्सिनिन 4 एरिन सुगर ट्राइजोल कॉन्जुगेट, लुक हियर, एजाइड है और ये अल्काइन है दिस मॉलिक्यूल। नेक्स्ट, खत्म कर लिया आगे। Why click chemistry? 100% reliable, quick and have wide scope, functional group tolerance, ये सब बड़े इंपॉर्टेंट पॉइंट हैं। Aqueous condition, shorter reaction time, high determinism, high yield, high purity, no chromatography required, regio and stereo specificity. Next, conclusion. Stepwise non-concerted mechanism account for 1.5 regio selectivity. Most popular among the synthetic methodology for the synthesis of triazole and related structure. Click chemistry has uniquely become a established tool for the construction of smaller to complex architecture. Most modular approach that uses only the most practical and reliable chemical transformation. Click chemistry works reliably in both small and large scale equation. Click now can be used a noun, click, verb clicking, adjective clickable, click. Next, thank you for patience. गला सूख गया, मैं रोजे से हूँ इसलिए मुश्किल हो रहा है मुझे बोलने में।
Thank you, Professor. Any question from audience? This is a very nice lecture.
Thank you. कॉपर 2 कभी नहीं यूज करेंगे, कॉपर 1 ही। आप कॉपर सल्फेट लेंगे, आप वो सोडियम एस्कार्बोनेट कॉपर सल्फेट कॉपर 2 में है, वो कॉपर 1 में कन्वर्ट कर देता है। हाँ, वही काम करता है, कॉपर 2 नहीं, कॉपर 1। हाँ, रुदेनियम। हाँ, अब मैंने बताया ना, जितने मैंने मेटल प्रोसेस लिए हैं सबके लिए अलग अलग मैकेनिज्म है। तो इस लेक्चर में 30 मिनट जिन्होंने कहा था कि 30 मिनट का टाइम मिलेगा, उस हिसाब से हमने 42 स्लाइड लिया था। तो अब मैकेनिज्म हम इसको बिना देखे नहीं बता सकते। मेरा सब्जेक्ट ये नोबेल प्राइज टॉपिक है, सो आई कैन नॉट बी मास्टर ऑफ दैट, यू शुड रियलाइज दिस। थैंक यू वेरी मच।
Presented. There are some questions are there.
हाँ, ऐसा है, रेडियो सिलेक्टिविटी मतलब कि एक रिएक्शन में एक से ज्यादा प्रोडक्ट, 2 या 3 प्रोडक्ट बनेंगे, तो कौन मेजर होगा कौन माइनर होगा, यही पता लगाना रेडियो सिलेक्टिविटी है। नहीं समझ पाया?
Is there any question? Okay. Thank you, Professor.
थैंक यू सर, थैंक यू सो मच। आप लोगों को बताते हैं मेरी एमएससी और बीएससी मैंने इन्हीं के अंडर में की और मेरे पीएचडी थीसिस पे इन्हीं का साइन है। थैंक यू सर।
इसके बाद लंच ब्रेक है। ये लेक्चर सिर्फ 15 मिनट का होगा, उसके बाद लंच होगा। स्पीकर हैं डॉक्टर A.N. शुक्ला, ये हैं बॉटनिकल सोसाइटी ऑफ इंडिया से सीनियर साइंटिस्ट हैं। इन्होंने पोस्ट डॉक NBRI लखनऊ से किया है। He has published 4 books till now. He has to his credit 75 research papers in international reviewed journals. He is additional director in MOEFCC New Delhi. Dr. A.N. Shukla.
Thank you very much. I hope I'm audible to everyone. Am I audible to everyone?
Yes, sir. You are audible.
And my slides are visible?
Yes, sir.
Okay, so I will be very quick. Right now, I'm working as the Additional Director, Ministry of Environment and Climate Change in New Delhi, not in Botanical Survey of India. Earlier, I was scientist in GSI.
Sir, your slides as well as you both are not visible.
Slides are not visible?
No, sir. You are doing? Yeah, now it's visible.
Okay.
You and your slides both are visible now.
Now it's okay?
Yes, sir.
Okay.
Okay, thank you. So thank you very much, Principal Professor J.P. Pandey, convener of the conference, Professor R.K. Singh, Dr. Sadhguru Prakash Ji, organizing secretary, Dr. Sunil Kumar Mishra, and respected Dr. A.K. Verma, honorable member UPSC. So I am not a chemist because the conference is being organized for chemistry student, chemistry scientists, and chemistry professors. I'm a botanist basically. So last year in ministry, I am looking after some policies like and some international convention like CBD and Biological Diversity Act. I'm not involved in research rightly. So last year in 2022, the very important Kunming-Montreal Global Biodiversity Framework was adopted by the Convention on Biological Diversity in COP15 at Montreal. So I will be very brief on the issue, which is very important for biodiversity conservation for all streams, for chemistry, for the people of geology and botany. So as everyone knows that CBD is very important convention for biological diversity, and India is also signatory of this convention, and it has 3 goals: conservation, sustainable use, and fair and equitable sharing of benefits arising from the use of genetic resources. These are the 3 main goals. On the basis of 3 goals, India has a very robust act that is called Biological Diversity Act, which was enacted in 2002. So in the background I'm giving, because the COP15 presidency was with China, and due to COVID, China refused to host the conference. So it was shifted to Montreal, as CBD has their headquarters at Montreal. That's why it is called Kunming-Montreal Global Biodiversity Framework. So earlier it was decided to host by China in Kunming. The framework is called Kunming-Montreal Global Biodiversity Framework, and and it is very important as pointed out by because as everyone knows that earlier in 2010, very important HE targets, 20 targets was adopted by the CBD, but the difference between HE targets and KMGBF, that is, in HE target, all the targets were aspirational and visionary because there was no numericals, no measurables. In KMGBF, there are certain targets had numerical values. They are measurable, and they are very important for biodiversity conservation. And in KMGBF, it has a vision by 2050. Biodiversity is valued, conserved, restored, and wisely used, maintaining ecosystem services. So this is the vision by which is going to be achieved by 2050, and it has a mission for 2030 to take urgent action across society, and the mission by 2030, which is achievable, and this is a measurable also. And this is a news cutting. And in confirming Montreal Global Biodiversity Framework, urgency to act, like in the world, and the CBD 196 countries are signatory of the CBD. India is also signatory. So biodiversity loss, it is one of the one of the major threat to biodiversity, and is the biggest risk we face. Last decade, effort did not produce expected result today. So last chance to put system back on track. That's why it is called the goals for 2050 and targets for 2030. And as everyone knows that IPCC is for climate change, and the same line IPBS, the independent intergovernmental body established by the states to implement the science policy interface for biodiversity and ecosystem services. So, on the basis of the assessment done by the IPBS, the some of the agenda goes to COP, CBD COP. So, on the basis of the assessment done by the IPBS, the agenda discussed during the COP of CBD. So during the 2019 to 22, there are 4 or 5 working group meetings, different countries. Like working group 1 was held at Nairobi, then second was Rome, third was Geneva, fourth was Nairobi, and fifth was in Montreal. Fortunately, I am looking after the CBD, and I was part of the Indian delegation at Montreal, headed by our honorable minister. In extensive negotiations done by the 196 countries, finally the decision was adopted as KMGBF. So before the COP, there were 5 working group meetings in different countries. And in COP 15, major outcomes at like Kunming, Montreal Global Biodiversity Framework, and monitoring framework, mechanism for planning, monitoring, reporting, and review decision, resource mobilization. You see, this is one of the important decisions done by the CBD. The targets are very much measurable, as I have already spoken. So from where the resource comes. So the developed countries, they have committed to fund to developing country to achieve the targets. So this is one of the major decision taken by the COP 15, resource mobilization, capacity building, and DSI, digital sequence information on genetic resources. And there are 4 goals, as I have already spoken, the goal A, B, and C. Goal A, goal B, and goal C. This are the targets, and goal D, this is the additions of funding gap. The 700 billion USD is closed by ensuring adequate means of implementation are available. So all the developed countries they have committed to share the fund with all mega diversity countries, all developing countries. And these are the 23 targets adopted in COP 15. Like and they are based on the major themes like theme first, reducing threat to biodiversity. Theme second, meeting people's need through sustainable use and benefit sharing. And theme third, tools and solution for implementation and mainstreaming. So these are the main 3 themes of the targets, and the targets of 1, 2, 3, 4 are written here. And some of the important targets like target 30 by 30. What has been discussed in COP 15 that by 2030, at least 30% of the land and sea must be achieved, must be restored, or must be conserved. So this is a very important target for India also, as we have more than 24% forest area, and some of the areas which are potential to be declared as protected areas. And one of the important terminology like OECM. This is a other than protected area, other effective conservation measures, and this has to be implemented. We have already made rules, and we have already made the guidelines for declaring OECM, so that we can achieve 30% conservation of our sea, land, by 2030. And India is also signatory of High Ambition Coalition, that is for also 30 by 30. And when we are implementing the targets, in when we are achieving, we are doing anything for conservation, we have because these are all targets are cross cutting, and we have identified 24 ministries which are dealing with the conservation of biodiversity, and we are sharing all the indicators which are going to be reported to CBD, to Ministry of Agriculture, Ministry of Health, Ministry of Rural Development. All across 24 ministries have been identified, and we have identified 257 complementary indicators for thematic or in-depth analysis of each goals and targets that could be used to supplement the proposed headline indicators where appropriate. So we had 4 regional workshops across the country, in Northeast, in Central India, in South, and in Jammu and Kashmir. We invited all states stakeholders, major stakeholders of central government, to discuss the targets which are going to be implemented and which are going to be reported to CBD. And I have just discussed that some of the targets which are measurable, which have the numerical value, like target 3, this is 30 by 30, target 6, the invasive alien species at least 50% reduction. I am informing you all, August gathering, that India did not support the numerical value, because we don't believe on the values, but it is global targets and has been adopted by the CBD. We have to have implement, and we have to have report to CBD. So we have selected some of the indicators, like in invasive alien species. We were agreed on the introduction checkpoint of introduction of invasive alien species, but we could not agree on 50% reduction of invasive alien species, because this is the policy matter, and this is a state matter. So we were a little bit not in agreeing. Reduction of chemicals and pesticide, climate change impact, and elimination of substances harmful for biodiversity. So as we are giving subsidies for agriculture, we are giving subsidy for electricity. So for this also, we were not in position to agree with the convention. But as I have already spoken, that this was agreed by the word and now adopted. So we have to report in some way. And resource mobilization. Every year now, just last month we received 115 crore for recruiting and for conservation of biodiversity. So we are going to have a major project for this. And road ahead, what we are doing now, we are now updating our NBSAP, National Biodiversity Study and Action Plan. So by the end of April, we have to submit to CBD. So we have already done the MASHU work, and this April 5th, we are going to have an interministerial workshop, so that all ministries which are major stakeholders for biodiversity, they can give their inputs, and after that, we'll submit to CBD. So this is the key steps for preparing and updating India's National Biodiversity Study and Action Plan, updating existing NBSAP, 23-member high-level working group. We have already constituted. Emphasis on consultative process. We have already had 4 consultative workshops in regional, state, and thematic themes. Setting up national targets, state and action points is in advanced stage. The deployment of draft biodiversity monitoring framework. These are in process. So thank you very much for giving me time, and I thank you all for patience hearing. Thank you very much.
Thank you, Dr. Sukla. Any question from audience? Okay, Dr. Sukla, I want to know what is related with this pollution and population with biodiversity.
Just would you like to? I'm not hearing. Can you just? Pardon?
Just I want to know what is relate this biodiversity with the pollution and population.
Sir, as this is one of the target of this KMGVF, the reduction of pollution also. We have a target 8, which are related to reduction of pollution, and we can relate how to reduce pollution to accelerate the plantation in forest area or in as agroforest in agriculture field also. It is not directly related to pollution or in population, but some way it's the indirectly related with the pollution, not to population as
Okay,
such.
okay. Thank you. Thank you,
Thank
sir.
you.
Thank you, Dr. Professor. Okay. Thank you.
अब जितने भी आमंत्रित अतिथि हैं, आप सभी लोग मध्याह्न भोजन के लिए पुस्तकालय सभागार की ओर प्रस्थान करेंगे और बच्चे जो हैं, जो स्टूडेंट्स हमारे हैं, ये लंच के लिए सूचना केंद्र पहुंचेंगे।